Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I'm your biology educator. Yes guys, so we are back once again in Re-NEET Bounce Back series, and we are going to complete zoology of class 11 today. We have already completed part 1 yesterday, and this day is for second part, and today guys we are going to discuss human physiology. Complete human physiology will be discussed along with NCERT reading and discussing from 0 to hero level. Good evening all of you. Give me a thumbs up that you're all set and ready with your NCERTs and your notes. Good evening. And also let me know that did you, those of you who have watched the the marathon yesterday, later on like the recorded form, have you done all the homework? And even those who watched it live, have you done the homework well? Because it's very important that now when we have got this opportunity, we don't leave any loophole. We are going to get better this time and we are going to crack NEET this time, no matter where we are or where we were. Good evening all of you. Yes, quickly like, subscribe and share with all your friends, and I am going to start. And also guys, in the beginning itself I want to tell you that all those who are preparing for NEET, Unacademy is by your side guys. You will be getting 700 plus hours of lectures, 30 plus tests, almost for free, just ₹1. So you have to subscribe for this Prime membership. You'll be getting all this on Unacademy app. Is that clear? With 15,000 plus practice questions in AIMT All India Mock Test Series. It is very important. Not even a single day should be left without taking the test. Not even a single day. And also guys, I have asked you to list down all the shortcomings that you felt during writing your NEET 2026 exam. Yes guys, have you done that? How many of you have done that? Please let me know. Hi, good evening. Ma'am, explain line by line. Naveen, that's what happened. I subscribed. Exotic, I'll please contact the tech department. Clear? Yes, I'm very fine. Volume is clear. All right, superb. And guys, those of you who are going to prepare for NEET 2027, I'm sure you have heard the news. It's going to be CBT, that is computer based test. So of course our preparation is also going to be according to that. And see, the students who are taking online tests are going to have an upper hand here, because we'll be already used to that. Those who are like, of course most of us, we try to download the paper and we do it paper pen style, right? Fine, that is good. But now since the paper style has changed for NEET 2027, it is very important that you take these online tests. Okay, and so for 2027 guys, this is the Phoenix 2.0 series, which you can get for under 4,000 for the complete year. The batches are going to start from 20th May. I'm going to start the batch, and you can get this for just under 4K, that is 3982, by using the code TR Live, Tina Randhawa Live, right? TR Live and the Phoenix 2.0. And guys, by taking this batch, it's not that you can just attend my lectures. You can choose your educator, you can choose your batch, and you can go ahead with that. So there'll be different batches. This is the English batch that I'll be starting with my teammates, and it's starting from 20th May. And with that, I am going to start, yes, the first lecture. Ma'am, Telegram channel, I have already guys mentioned in the messages also. Those of you who have messaged ma'am Telegram channel, still I will tell you very clearly. Yes, so you have to join me on Tina Ma'am Biology on Telegram channel. Just write down Tina Ma'am Biology and you will find all the notes I have already uploaded. Tina Ma'am Biology. Clear? So this is the Telegram handle and you will get all the notes. I've already uploaded the notes for the yesterday's lecture, and we'll do the same for today's lecture also. Now quickly give me a thumbs up. You're ready with your note making things, and we can go ahead. Quickly let me know. Yes. Shall we start? Yes, please. All right. Perfect. Now guys, we are going to start with human respiratory system, that is breathing and exchange of gases basically. So what does this first paragraph of this human physiology thing says? What does it say? Let's see. Okay. Now it says, first of all, see, as you read, you have read earlier, oxygen is utilized by all the organisms. This is this to derive energy. So we utilize oxygen to derive energy to perform various activities, and CO2 is harmful, which is released and let out. Now there is a proper system to release the carbon dioxide and inhale oxygen. So this is the chapter for that. Also it says there are two different terms. See, breathing, which is commonly known as respiration, but we know that these are the two very, very different things. These are two very, very different processes. Now what is breathing, guys? Breathing is only about inhalation and exhalation. You inhale, you exhale. That is breathing. You also call it ventilation, right? Now, but breathing is just the first step of respiration. Now, when we say respiration, it means breakdown of food to release energy. Is that clear? So, we'll be talking about all the steps which starts from breathing and moving on to and going up to release of energy, that is respiration. Is that clear? So, respiratory organs, the mechanism of breathing, all is going to be described in this chapter. Moving on to the respiratory organs first of all. All right, here. Now see, mechanism of breathing varies from different organism. For example, in invertebrates like sponges, coelentrates, flatworms. See, I've already learned it yesterday. All these organisms, they don't have a proper respiratory system. So they do that from their body surface. The entire body surface is used for the breathing or the exchange of oxygen and carbon dioxide, and the method used is simple diffusion. First point, right? What about earthworms? Earthworms are going to use this moist cuticle, right? So earthworm shows what type of respiration? It is cutaneous. We have learned that in animal kingdom already. Then insects have a special network of tubes which is called trachea. So they have tracheal system, yes, and special vascularized structures called gills. And all those organisms who respire through gills, they show branchial respiration. You already know which are those. Even some arthropods do, right? Gills, book gills. Then our fishes, they are all going to use gills. Then aquatic arthropods, already told you. Some mollusca, they have feather gills, remember? And of course the fishes, they all use gills. But we are talking about the lower organisms. So vascularized bags, which are known as lungs, they are used by the terrestrial forms. So which all terrestrial forms are going to use them? So yes, all the terrestrial forms. Now what do we call it? The respiration which is occurring through lungs is known as pulmonary. We have read all these things in animal kingdom. In case you have missed that, don't forget to watch that again. Then amongst the invertebrates, guys, invertebrates, that is fishes, they use gills. Amphibians, reptiles, birds and mammals, they respire through lungs. But we have seen some exceptional cases, like in case of frog. Frog can respire through skin. Frog can respire in the larval stage, it can respire through gills. But as an adult, when it is underwater or it is hibernating or estivating, it uses skin. Yes or no? How many of you do remember that? How many of you remember that? Is that clear? Tina, ma'am, biology, you guys, you have to put M-double-A-M. Ma'am, okay? All right now. And then, talking about frogs. So, frog will use mouth, that is buccopharyngeal respiration, and lungs, that is pulmonary respiration while on land. So, amphibians like frog, see, frog, it can respire through moist skin, cutaneous respiration also. And when does it do that? Underwater and during hibernation and estivation. Please tell me these points are clear. We are going to keep revising what we have learned in the previous chapters as well. Is that clear? Is that clear? Now, human respiratory system. Human respiratory system starts with the external nostrils. These are the two external nostrils open and leading into the nasal chamber through the nasal passage. So first we have nasal passage, This is nostrils, nasal passage, moving into the nasal chamber, and then this chamber opens into the pharynx. So you should know the correct pathway. Now this is a common passage for food and air. Yes or no? Now pharynx opens through larynx. Now through pharynx it goes into the larynx, and larynx will open into the trachea. Now larynx, guys, we know is a cartilaginous box. We also call it sound box because it will help in the production of sound. Is that clear? Now it is a cartilaginous box. This is what you have to remember. Now during swallowing, glottis, which is covered by the thin elastic cartilaginous flap called epiglottis. Now what is epiglottis? See, these are the nostrils, right? These are the nostrils. So nostrils basically lead into the nasal passage and then the nasal chamber, right? And see here. So this is the buccal cavity. This is the roof of the buccal cavity. So this is the nasal chamber. These are the nostrils, external nares or nostrils. This is the nasal chamber leading into the common passage, which is called pharynx, and pharynx leads into the what? It leads into this trachea. Now in the beginning of trachea we have this voice box or the larynx. Yes or no? Right? And now this opening, see here, the opening of trachea is known as glottis. The opening of trachea is known as glottis, and glottis is guarded by this flap-like structure called epiglottis. I hope all these points are clear to you. And now epiglottis is also made up of cartilage. It is also made up of cartilage. Are these points clear? Now, what is the purpose of epiglottis? Epiglottis is going to prevent the entry of food into the trachea or into the larynx. Now, this trachea is a straight tube. This trachea, I'm going to elongate this one. This trachea is a long tube which is marked by or which is surrounded by these C-shaped cartilaginous rings, right? And then it breaks off or branches into the two bronchi. Now these bronchi will enter the lungs. These bronchi are going to enter into the lungs. Now the point to be noticed here is, where does it divide? See, trachea is going to divide, it divides at the level of 5th thoracic vertebra. Please tell me that these points are clear. So at this point on the dorsal side, your 5th thoracic vertebra is present. Please tell me these points are clear. Yes, please. Please tell me these points are clear. No, the voice is absolutely fine. Okay, all right. Now what happens? So it breaks off into the right and the left bronchi. Each bronchi will be it will undergo repeated divisions to form secondary, tertiary bronchi, and then the bronchioles, ending up into the thin terminal bronchioles. Now see, all these structures, they are supported by incomplete cartilaginous rings. Now, each terminal bronchiole gives rise to a number of thin, irregular, walled and vascularized bag-like structures called alveoli. So, after the terminal bronchioles, terminal bronchioles will lead into the alveoli, and alveoli, as we know, is the unit of lungs. Is that clear? Now, it is written, see, we have two lungs and they are covered by a double-layered pleura. So, pleura is present here. So this is the pleura. So the double layered pleura, this is one, and similarly we have two. So it is a double layered pleura. So pleura, these are the protective coverings, and the space in between, guys, this is all filled with the pleural fluid. Is that clear? This is all filled with the pleural fluid, and this is where it is written, pleural fluid between them, which reduces the friction on the lung surface. Is that clear? Outer pleura. So see, this being the outer pleura and this being the inner pleura. Please tell me that these points are clear. Outer pleura is in close contact with the thoracic wall, while the inner pleura is in contact with the lungs. Please tell me if there is any doubt in this line. So this is the diagram which is given in your NCERT. So after understanding, ultimately we have to come down to NCERT. So see here how epiglottis has been shown. So this is the epiglottis. Okay, this is the epiglottis. Now this is that voice box called larynx. Do you see this long tube called trachea, the windpipe? And it is marked by, it is surrounded by these cartilaginous rings. Remember, these are C-shaped rings. We call it C. Ring which is C means it is incomplete. Incomplete cartilaginous rings. So these rings are only found on the ventral side. They are only found on the ventral side. Why? Because on the dorsal side we have esophagus. If there will be cartilaginous ring on the dorsal side, your esophagus cannot show peristalsis. So it is only on the ventral side. Do you see? So all throughout, even in the bronchi, these will be present. Then bronchi into all these primary, secondary, all of them will have these cartilaginous rings. Are these points clear? Yes or no? And do you see that ultimately the terminal bronchioles, they are leading into these structures which are called alveoli. These structures are called alveoli. So this is how they have shown the pleural membranes, the one in yellow. These are 2 in number and they are filled with the pleural fluid, and these are the bronchi. Now look at the location, the position of your lungs. So do you see? The lungs are protected by ribs on the outer surface. So lungs are placed in the thoracic cage. They're protected by the rib cage. So these are the cutout of the ribs that have been shown here. These are your lungs. And on the lower side, on the inferior side, we have diaphragm. Now without diaphragm, your lungs cannot move. We will see the significance of diaphragm during breathing. Please tell me these points are clear to all. Please tell me all these points are clear to you. Yes or no? Are these points clear to you? How to focus? Raman, this is very easy. See, we know that you have read NCERT multiple times. We have already made many sessions. We have you have already recorded many videos on these sessions. But why are we taking the same sessions, the same book again and again live, so that you can sit with us and revise it? So this is the best way to focus. Study with us. That is the best way to focus. If you can't focus now, then I think I am helpless, right? Now see, so we are reading things with you. How else do you want to focus? Now see, the inner pleura is in contact with the lung, already read. The part starting with the now this is very important, very important. So the overall respiratory pathway that you've read is divided into 2 components. One is the conducting part, and then we have the respiratory or the exchange part. Now see very carefully, from external nostrils to terminal bronchioles, it is the conducting part. Okay, and then alveoli and their ducts, these constitute the respiratory or the exchange path. Now the name itself tells you what are they going to do. So the conducting part, the function of conducting part is to conduct the air. Second, it also humidifies. Now see here, the conducting part, what is the function of conducting part? It, number 1, it transports the atmospheric air to alveoli. Number 2, it clears it from the foreign particles, because you already have hair and mucus here. So hair is going to act as a air filter. So they clear the air. One is transport, second is clear, third function is to humidify, that is adding moisture to the air. In case the outer air is not sufficiently moistured or humidified, doesn't have sufficient water content, your conducting part is going to add moisture to that. And also it brings the air to the body temperature. That means thermoregulation would also take place. These are the 4 functions of the conducting part. 1, it is transport. Second is clearing it from the dust particles or the foreign particles. Third is adding moisture, humidification. And fourth is thermoregulation. Are these clear? While it says the exchange part, the only function it is for actual diffusion of oxygen and CO2 between blood and atmospheric air. So as the name says, exchange is only going to exchange the gases. Please tell me these points are clear. So you should be very, very thorough with what are the parts coming under the exchange. That is only alveoli and the ducts. Alveoli and their ducts constitute the exchange part, while everything before this, that is external nostrils till terminal bronchioles, they constitute the conducting part. Please tell me these points are clear. Please tell me these points are clear, right? Now moving on to the next, that is lungs. So where are lungs situated? Lungs are present in the thoracic chamber, already told you. And this thoracic chamber is, guys, airtight chamber. This is an airtight chamber. Now, how is this chamber formed? So we have ribs, we have vertebral column, we have sternum, and on the lower side we have diaphragm. See, thoracic chamber is formed by, on dorsal side we have vertebral column. Told you, on the back side we have vertebral column, on the ventral side we have sternum. Ventral side we have sternum. On the sides, that is laterally, it is formed by ribs, while on the lower side, guys, there is a dome-shaped diaphragm. Please tell me that these points are clear. And all these four structures constitute the airtight chamber or the thoracic chamber. 1, that is 2, that is 3, and that is 4. I'm sure you have marked these things in your NCERT already. In case you have missed out anything, please mark it along and number it. Numbering is very important. It's very important that you learn the things in the list form so that you remember, okay, 1, 2, 3, 4. Or it should be very crystal clear in your head. Is that clear? Now, the anatomical setup of lungs is such that any change in the volume of thoracic will be reflected in the pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. Now see, remember, breathing is the breathing process totally depends upon the pressure difference. So we know this is the basic principle of physics, that air moves from high pressure to low pressure area, right? So if you want the air from atmosphere to enter into the lungs, so basically it has to be low pressure in your lungs. What is it saying? You cannot directly change the lung pressure, but what can you do? You can manipulate the volume of the pulmonary cavity. Are you getting that? Now we'll understand more about it when we come directly to breathing. Now this was a direct question of 2026, yes or no? Direct question of 2026. So you have to arrange it in the correct sequence. I'm telling you again and again, NCERT is loving to give these sequence based questions. So this was the question. The first step is breathing, also known as pulmonary ventilation, where there is direct exchange between atmosphere and the alveoli, right? Second is the diffusion of gases across the alveolar membrane, right? That is between alveolar membrane and blood. Then there is transport of gases by the blood. The blood is going to carry it. Then finally diffusion of gases it's going to occur between blood and tissues, and finally utilization of oxygen by the cells to produce energy. So this is going to be a cellular respiration, about which we are going to discuss tomorrow in botany part. Please tell me these points are clear. Please tell me these points are clear. Yes or no? Chemistry class is at 8:30. Which chemistry class, guys? My class is going to be, ma'am, yesterday's session PDF wasn't shared. Mauli, I've already done that on my session, also my Telegram channel. That is Tina ma'am biology. Okay. And even Wasim sir has shared it on his official channel. Now, coming on to the first step. So we have already seen there are 5 steps. We'll be discussing all the 5 steps one by one, starting with the process of breathing. See, breathing occurs in 2 steps, 2 phases. One is inspiration, second is expiration. Inspiration is inhalation, taking in air, and expiration is breathing out of air. Now, the movement of air, this is going to occur between lungs and atmosphere. This exchange is called breathing. Breathing is also known as pulmonary ventilation, we have just read. Now see, this can only occur, inspiration occurs if the pressure within the lungs, intrapulmonary pressure, is less than the atmospheric pressure. Yes, I told you already that air moves from, because air moves from high to low pressure area, high pressure to low pressure area. Now, if you want inspiration to take place, for inspiration, that is inhalation, where is the high pressure going to be? For air to move into the lungs, so lungs will have low pressure area. While during expiration, if you want the air to come out of lungs, that means your lungs should have higher pressure. So lungs have high pressure. So intrapulmonary pressure, that is pressure inside lungs, should be low. Is that clear? Is less than the atmospheric pressure. And there is negative pressure. We call this as negative pressure. Negative. Right? While in expiration will take place only if the intrapulmonary pressure is higher than the atmospheric pressure. So it's going to be positive here. Is that clear? Now, as already been said, you cannot directly change the pressure. But what you can do, you can change the volume. So there are certain structures which are involved in doing so. What are those structures? Diaphragm, specialized set of muscles which are known as external and internal intercostal muscles. Inter means between, costa means ribs. So where are they present? Between the ribs. Right? So these are the two main structures which are going to help in changing the pressure. So now let me tell you from this diagram. Yes. So this diagram represents inspiration because air is entering the lungs. So if you want air to enter, so there has to be negative pressure or low pressure in the lungs. How do we generate the low pressure area? See, understand. So we know that pressure and volume are inversely related. If you want there has to be negative pressure, then you have to increase the volume of the thoracic cavity, right? So see, ribs and sternum are going to raise. How are they going to raise? Because of the movement of external intercostal muscles. External intercostal muscles. Is that clear? They will contract. Contraction of the muscles, external intercostal muscles, will lead to rising of ribs. So see, when you inhale, so your ribs and your sternum move outward, forward, and they get the structure. This thoracic cavity is going to, the volume of the thoracic cavity is going to increase. So because of, now see here, because of ribs, the volume of thorax increases in the, this is ventral, this is dorsal. So it increases in the dorsoventral axis, yes. So you have increased the volume by raising the ribs and the sternum. But when the diaphragm contracts and it becomes flattened, now you are increasing the volume in which direction? You are increasing the volume. When see, diaphragm moves downward, it becomes flat from dome shape. So you are increasing the volume. So the volume increases in the anterior-posterior axis. So now this is already a PYQ. You should know that which structures are going to increase the volume in dorsoventral axis and which are going to increase the volume in the anterior-posterior axis. And now, after the volume has been increased in all the four axes, what will happen? Since the volume has increased, see, volume has increased, pressure will automatically decrease. So volume increases, so here what happens? The volume increases, so pressure decreases. And therefore air goes in. Is that clear? Please tell me these points are clear to you all. These points are clear. So dorsoventral axis, the thoracic sternum causes increase in the volume of dorsoventral axis, and similarly the increase in the pulmonary decreases during the intrapressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs. We have discussed it all. That diagram is very, very important. Moving on to the second phase of breathing, which is exhalation. Now see what is going to happen in exhalation. So ribs and sternum, they return to the original position. That means they move backward. backward and inwards, right? Now, when they move backward and inward, this is going to decrease the volume again. In which axis? Dorsoventral axis. Yes or no? So this is the dorsoventral axis. So volume is going to decrease here. Now, the diaphragm moves backward. Diaphragm relaxes and arches upward. That means it becomes dome shape. So this shape is called dome shape. Yes, so this also decreases the volume, but in anterior-posterior axis. Just the opposite of what happened during inhalation, in anterior-posterior axis. Yes or no? So ultimately, the overall volume of the thoracic cavity is going to decrease and the pressure within the lungs is going to increase. The pressure here will become positive. It will become more. High pressure will develop here. So, of course, air is going to move out. Air is going to move out. So, exhalation takes place. Please tell me all these points are clear. Please tell me all these points are clear. Now, there is this important line written here that we have the ability to, just a second. We have the ability to, guys. Yes. So we have the ability to increase the strength of inspiration. See, normally we are inhaling, it's a silent breathing, but I can breathe like this and I can exhale also like that forcefully. So whenever there is forceful breathing involved, see, increase the strength of inspiration and expiration can be increased with the help of some additional muscles in the abdomen. Additional muscles. So basically we are going to use the abdominal muscles. You can feel the stretch also. When you inhale like that with a stretch, with stronger inhalation, you feel the stretch in your abdominal region, right? Also your internal plus your internal intercostal muscles are used. Internal intercostal muscles. See, remember, during normal breathing, it is the external intercostal muscles, while during the forced breathing, this is the forced breathing we are talking about, forceful breathing, it is the internal intercostal muscles and the abdominal muscles that are used. Please tell me these points are clear. Then it says that healthy person breathes around 12 to 16 times per minute. So guys, this is your breathing rate. This is your breathing rate. Please read the question well, whether they are asking for breathing rate or the heart rate. Heart rate is between 70 to 80 or on average 72 times per minute. You don't breathe 72 times. That will be like another situation. So you breathe 12 to 16 times per minute. Now, how much volume you inhale or you exhale during normal breathing or during forceful breathing, that can be measured through a special device known as spirometer, and this is helpful in clinical assessment of the pulmonary function. So why do we use that? It is not for fun, of course, that okay, let's check how much volume have I inhaled. It has a clinical significance. So we can check your lung health, lung capacity by the help of this spirometer. Please tell me these points are clear. Yes, guys. Now this topic is super important. Super important. Now 2026 NEET forgot to ask question, but otherwise you generally see question from this topic. Pulmonary volumes and capacities. So first of all, let me differentiate between the volume and capacity. Remember, volume is going to be a single value. Is that clear? While capacities is going to be multiple values. You will understand how. Now see, coming on to tidal volume. So you have to see the keywords. What is tidal volume? Tidal volume is the amount of air or volume of air that you inhale or exhale during normal breathing. So what is the keyword here? Normal respiration. Whether you inhale or you exhale, but it has to be normal breathing. And what is the value here? The value is 500 ml per breath. Is that clear? So now since we have read that how many times do we breathe? 12 to 16 times. So 12 into 500, 6,000, and 16 into 500, 16 into 5 is 80, right? 8,000 milliliter of air per minute. Please tell me these points are clear. Yes, guys, please tell me these points are clear. Yes or no? Are we all focusing? Are we all focusing? Yes or no? Yes, please. Are we all focused? Anyone who is feeling the distraction yet? Yes, please. Are we all here? Perfect. Perfect. Now see, the value of tidal volume per minute, value per minute is this, but per breathe is 500 ml. You get these questions in the form of match the following. You have to match the value. Also, you can get it in the form of definition, which of the following is the correct definition. And if NTA or any paper setting agency uses a little brain, they can also give you numericals out of this topic. Now listen, inspiratory reserve volume. Inspiratory reserve volume. What is the key word here? So it is the additional volume of air that is inhaled during forcible inhalation. So see, sorry. So if you inhale normally, that is the tidal volume. Inhale, exhale. 500 in, 500 out. But 500 in, and on top of that, when you top it up with an extra amount of volume, extra volume of air, that is called inspiratory reserve volume. Is that clear? Now, how much extra you can inhale? That is 2500 ml to 3000 ml. Please tell me these points are clear. Please tell me these points are clear, right? And then we have expiratory reserve volume. Expiratory reserve volume. Now in expiratory reserve volume, this is the additional volume of air other than tidal volume. See, you exhale 500, but after 500 also, beyond 500 also, you can still exhale forcibly, right? So it is the additional volume of air that we can expire by forcible expiration. And how much extra you can expire? That is 1000 to 1100 ml. Now please note down this key point. During inspiratory reserve volume, it is the forcible inspiration. But remember, hashtag exhalation is normal. Inhalation is forcible or maximum, you can say forcible. This is normal. While in this case, inhalation is normal, exhalation is forcible. Are these points clear? Please tell me these points are super clear to you all. Please tell me these points are super clear. Yes? Then moving on to the next value. We have residual volume. Now, as the name says, residue means what is left behind. So, no matter how forcibly you exhale, there is some amount of air which will always remain inside the lungs. So, see here, it is the volume remaining in lungs even after forcible expiration. No matter how forcibly you exhale, you can never empty down your lungs. So, this value is between 1100 ml to 1200 ml. Are these points clear to all? Please tell me. This amount will never come out. Now coming on to the capacities. So talking about inspiratory capacity. Inspiratory capacity is the total volume of air that you can inhale. So that is normal, TV and IRV. So after normal expiration, the value, see, after normally exhaling the air, total volume of air person can inspire. That means forcibly. So inhalation is forcible while expiration is normal. So this is the total, because you take 500 and on top of it you take 2500 to 3000 ml. So that together becomes inspiratory capacity. Similarly, we have expiratory capacity, guys. So this is TV plus ERV. So in this case, there is normal inspiration, but expiration is forcible. Is that clear? But when I'm talking about vital capacity now, first of all, maximum volume of air person can breathe in after a forced expiration. So see, here only inspiration was forcible, here only expiration was forcible, but in this case, both inspiration and expiration, both are forcible. Please tell me these points are clear. Yes or no? And it includes these three values, ERV, TV and IRV. Is that clear? So either you exhale forcefully and then inhale forcibly, or you inhale forcibly first and then exhale forcibly. Either way, it is going to be vital capacity. Please tell me that these points are clear. Yes? So you do it yourself, you understand, right? So I think this part of the chapter actually makes you do all these breathing exercises. Now listen, talking about the functional residual capacity, it is FRC. What does this mean? Residual volume is no matter how forcibly you exhale, there is some amount of air that will always remain inside our lungs. But the point is, we are not always exhaling forcibly, right? Normally we are exhaling normally, right? So what is the volume of air that remains inside your lungs when you are exhaling normally? So that is functional residual volume. So it is the air that remains in the lungs after normal inspiration. See this. And it is ERV plus RV. So ERV is something that you can exhale, but RV will never come out. So normally it is ERV and RV that together stays in your lungs. That is the functional residual capacity. And last not the least, guys, we have total lung capacity, TLC. It is the total volume of air accommodated in the lungs at the end of forced inspiration, and this will include all the four components, that is RV plus ERV and TV and IRV here. Is it clear? Or you can simply call it vital capacity plus residual volume. So there are different ways in which you can write down the TLC. Please tell me all these points are clear. All these points are clear. Now, how many of you, who can tell me, if you can tell me this question, your fundas are right. Tell me which of the following, these volumes cannot be measured. Volumes and capacities which cannot be measured, that cannot be measured, just a second, cannot be measured by spirometer. Quickly let me know the answer in the chat. Yes. Which volume cannot be measured using the spirometer? Yes, please. Yes, guys. Yes, please. You cannot measure residual volume, FRC. Very good, very good, very good. Those of you who have written both the elements, so we cannot measure the residual volume, we cannot measure the FRC. One more. Is there one more that we cannot measure? Yes or no? Directly? Is there anything else that we cannot measure directly? Very good, alien. Excellent, excellent, excellent. Excellent. It is also, see, it is also the TLC. Now, these three values cannot be measured directly. They have to be calculated indirectly. Yes or no? Yes or no? Perfect, perfect, perfect. Ma'am, telegram please. Telegram. Yes, telegram. I have to give you my telegram. So, Tina, ma'am, biology. So, this is my telegram link and you will find all the notes there. I've already loaded yesterday's notes. I will be uploading some other PDFs also, like the PDF of NCERT book, where I've already marked the PYQs. I'll be sharing it with you. See, I've already taken the session for that before NEET 2026. I'll be doing that again, but if you want to read that yourself, I'll be sharing that PDF also in the same Telegram channel. Is that clear? I'll do that all for you. Okay. Additional word is not, ma'am, additional word is not, huh? Yes, guys. Is that clear? All three. Okay, perfect. Now moving on to, see, we have already exchanged the air between atmosphere, we have already exchanged the gases between air and the alveoli, right? Now, what are the primary sites? So air enters into the lungs, but what is that primary site of a gaseous exchange? So point is alveoli. That is why we call alveoli as unit of lungs. These are the unit of lungs because this is the primary site of exchange of gases between blood and air. So now I want to discuss the structure of alveoli first. Okay, I think I'll have to draw this. Now see here, and here comes your animal tissue also. So see, this is your alveoli, right? And these are your capillaries. There is a thick network of capillaries around your alveoli, right? Now see, for the gaseous exchange to occur, for the diffusion to take place, this wall has to be ultra thin. This wall has to be ultra thin. What type of cells are present here? The first type of epithelial tissue I'm going to discuss here, that is squamous epithelium, guys. This is the only type of epithelium which is capable of diffusion, right? And we must know that epithelial cells are characterized by the basement membrane. So there must be a basement membrane present here. Must be a basement membrane present here. Yes or no? This is the basement membrane. And then, now see, and then we have alveolar wall. This is the wall of alveoli, again lined by thin squamous epithelium. Please tell me that these points are clear. This is also thin squamous epithelium. Yes or no? Please tell me these points are clear. Now see, the squamous epithelium of capillary wall is known as endothelium. We call this endothelium. Now when the gaseous exchange has to occur, see, so gaseous exchange would be like oxygen enters here and carbon dioxide will enter here. So what are the three membranes that gases are crossing to move from one place to another? It is the squamous epithelium, the basement membrane, and the squamous epithelium of capillary, that is endothelium. So together these three, remember, together these three, they constitute what? The respiratory membrane. Please tell me these points are clear. Together all the three, they will form the respiratory membrane. And who can let me know the thickness of this respiratory membrane? Yes, please. How many of you know the thickness of this respiratory membrane? Quick, quick, quick. How many of you know the thickness? Guys, quickly concentrate here. How many of you know the thickness of this respiratory membrane? I'm waiting for the answer. And also let me tell you, there is this special layer of a phospholipid which is present on the inner wall of alveoli. And who can answer which phospholipid am I talking about? We have already discussed about it in the previous session, guys. You have to name this phospholipid, and it helps in maintaining the structure. It maintains the alveolar wall. Without this phospholipid, your alveolar wall will degrade. Maintains the alveolar wall. Yes, guys, who can answer? What is the name of that phospholipid? What is the name of phospholipid? Very good, guys, very good. It is nothing but lecithin. It is present here. So actually there are some other type of cells also which are present on the inner side, but we will not talk about it here. But lecithin is important to decrease that surface tension and maintain the structure of alveoli. Are these points clear? Are these points clear, everyone? Yes or no? Are these points clear to all? So now see, alveoli being the primary sites, exchange can occur between the blood and the tissues. Oxygen and CO2 are exchanged in these sites by simple diffusion. What is the method involved? It is by simple diffusion. by pressure concentration, pressure difference, solubility of gases as well as the thickness of the membrane are involved. So there are three factors which will be playing important role here. One is the pressure, second is the solubility, and third is the thickness. So I am asking you again, what is the thickness of the respiratory membrane? Nobody has answered. Nobody has answered. 0.2 to 1 micron meter. Are you sure? Yes, guys. Yes or no? Less than 1 mm. Less than 1 mm. Yes, true. Less than 1 millimeter. Is that clear? Now, talking about the pressure contributed by the individual gas is a mixture. Now, guys, whenever there is this movement of gases, each gas is going to follow its own pressure gradient. So, gas or air always moves from high pressure to low pressure area. So, if I'm saying oxygen is moving from alveoli to lungs, to the blood, why? Because this must be high here, high pressure of oxygen here and low pressure of oxygen here. If CO2 is moving here to here, high partial pressure of CO2 here and low partial pressure of CO2 here. Please tell me that these points are clear to all. So each gas is contributing partially to the total pressure. So we call it partial pressure. Is that clear to everyone? Yes. So these are some values. Now just looking at the value, you can clearly tell. So what would be the direction of movement of air? See here. So from atmospheric air, air moves into the alveoli. Is that clear? Yes. Oxygen moves from atmosphere to alveoli because in atmosphere we have higher pressure of air, what is 159, and alveoli has partial pressure of oxygen to be 104. So of course it is going to move from atmospheric air to alveoli. Yes or no? Okay. But carbon dioxide has higher pressure in the alveoli and lesser in the atmosphere. So carbon dioxide is going to move from alveoli to the atmosphere. Are these points clear? Can you see? So the underlining statement is that air moves from, or each gas follows its own partial pressure gradient. Gradient means difference. So if there is no difference, there will be no movement of air at all. Clear? So it moves from high partial pressure to low partial pressure area. Yes or no? And each gas moves independently. You must know the values of these. Is that clear? In deoxygenated blood, partial pressure of oxygen is 40, while that of carbon dioxide is 45. In oxygenated, it is 95, and pressure of CO2 is 40, while in tissues it is 40 and 45. You have to learn these values. Clear? Now, this diagram also given in NCERT, see here. So do you see? Now this diagram depicts how gaseous exchange occurs between alveoli and blood. We have seen that gaseous exchange between atmosphere and alveoli, and then how it moves from alveoli to blood and then blood to tissues. So two diffusions have been depicted here. See, why does oxygen move from alveoli to the blood? See here. So pulmonary artery, as we know, carries the deoxygenated blood, and we know the partial pressure of oxygen in deoxygenated blood, we have just seen it is what, 40, and partial pressure of CO2 in deoxygenated blood is how much, 45, right? Now see, this deoxygenated blood is brought into the lungs. Now oxygen is 104 here, while oxygen is 40 here. That is why oxygen is going to move into the blood from high to low. Then partial pressure here is 45, here it is 40. So CO2 will move from here into the alveoli. You see this exchange? Now when oxygen has moved into the blood, this blood becomes oxygenated, and that's what is carried by the pulmonary way. Now what is the pressure of oxygen in the oxygenated? We have just read 95 mm of mercury, of course. And partial pressure of CO2 from 45, it reduces to 40 because it has been taken out. Is that clear? Now this oxygenated blood through the whole systemic circulation is brought to the tissues, and we have seen, see, it is written here, this thing is written here, this thing is written here. Now what is the pressure of oxygen in tissues? We have just seen it is 40, while partial pressure of carbon dioxide in tissues is 45. So what will happen? Oxygen here is 95, here it is 40, so it will move from here to here. Here carbon dioxide is 45, here it is 40, sorry, yeah, 40. So carbon dioxide is going to move into the blood. Please tell me these points are clear to everyone. Please tell me these points are clear to everyone. Yes or no? Quick, quick, quick. All done? Is this clear? Is this clear? Can we move on to the next now? Now see, it is written gradient is present for CO2 in the opposite direction from tissues to blood and then blood to alveoli. So as the solubility of CO2 is this times higher, now this line is very, very important. This line is very, very important. Solubility of CO2 is 20 to 25 times higher than that of oxygen. We have read that during the exchange, it is not just the thickness, it is also the solubility that plays an important role. So see, the amount of CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure is much higher as compared to that of oxygen. So what is it trying to say? So if there is the same volume, if there is that same diffusion per unit difference in the partial pressure, if there is the same partial pressure difference, so the amount of CO2 that will transfer, if the difference is same in with respect to oxygen and CO2, the amount of CO2 that will transfer will be much higher because it has higher solubility. Is that point clear? So the diffusion membrane, it is writing, see, this is the diagram that we have already discussed. The diffusion membrane is made up of three major layers, which are thin squamous epithelium of alveoli, endothelium, epithelium of this, and the basement membrane. And together, see, together all of them, the total thickness is much less than 1 millimeter. Therefore, all these factors will be responsible for diffusion of oxygen from alveoli to the tissues and CO2 from tissues to the alveoli. See these structures, squamous epithelium, it has to be one cell thick. So it is simple squamous. So if it is in monolayer, it is simple. Similarly, endothelium is also simple epithelium. There is no other epithelium which can conduct diffusion other than squamous epithelium. I hope you all know that. Please tell me these points are clear. Please tell me. Yes, guys. These points are clear. Please tell me. Those of you who are spamming that it is low voice and sending me these stupid emojis, I'm warning you. Get focused here. I am already shouting. My throat is already hurting. But doesn't matter. It is our preparation time. Okay, is that clear? All right. Now, therefore, all the factors in our body, guys, those of you who are not able to find out the Telegram channel, just leave a comment. I'll share it with you personally. Don't worry. I'll share the link. You just leave the comment. Okay? No, just write down notes. I'll share the link. Don't worry. Thank you. All right. Are these points clear? Now, once there has been exchange between atmosphere and alveoli, then from alveoli to blood and from blood to tissues. Now talking about the story of blood. How is blood going to carry or transport these gases? Let's discuss that. See, blood is the medium for the transport. Blood is the medium for the transport of oxygen and CO2. Now blood is going to transport majorly two gases, oxygen and CO2, right? If we first talk about oxygen, so 97% of oxygen is transported by hemoglobin. And what will happen? Hemoglobin and oxygen are going to form oxyhemoglobin. Yes or no? This is going to form oxyhemoglobin. And one hemoglobin can carry how many molecules? 4 oxygen molecules. Yes? So it is going to be Hb4O8. This is oxyhemoglobin. How much percentage of oxygen is transported in this way? That is 97%. 3% of oxygen is going to be transported in the dissolved form, dissolved in plasma, because plasma has 90 to 92% water, so it will simply dissolve in water. Talking about carbon dioxide, only 23% is transported along with hemoglobin. Hemoglobin plus CO2 will form, please be careful, carb-aminohemoglobin, not carboxy. It is carb-aminohemoglobin. Yes or no? Clear? Now, this is just 23%. Then 7% of hemoglobin is dissolved in plasma. Just like oxygen is 3%, this is 7%. So it is just 30%. 70%, that is the major bulk, the 70% of CO2 is transported in the form of bicarbonate ions, guys, through plasma. Of course, it doesn't move in the form of these ions. So these bicarbonate ions are going to form sodium bicarbonate and potassium bicarbonate. And carbon dioxide is going to move in the form of these two salts through your plasma. So if I ask you this question, let me know in the chat, what is the total percentage of carbon dioxide transported by plasma? Yes, guys. What is the total percentage? Yes, guys. What is the total percentage of CO2 transported by plasma? Very good, Shoaib. Very good. It has to be 77. So total percentage, so total carbon dioxide transported by plasma is what? Is 77%. Please tell me these points are clear to you all. So this is just these are just facts. So see, it's written. So we write 23%. It says 20 to 25%. Is that clear? So 20 to 25% is through RBCs, by RBCs. 70% in the form of bicarbonate. 7% in the dissolved state. So total through plasma is 77%. Are these points clear? Please tell me. Are these points clear? Yes. Now moving on to the transport of oxygen here. See, hemoglobin. So how does this hemoglobin we have just talked about? See here. So hemoglobin, this hemoglobin which is transporting oxygen and CO2, how does this behave? So what is hemoglobin? Basically, hemoglobin we know is a red colored iron containing pigment present in RBCs. Yes, oxygen binds with this in the reversible manner. Remember that line. Oxygen and hemoglobin are going to bind in the reversible manner. This is very important, that if at one place, that is in alveoli, oxygen binds with hemoglobin, then at the tissue level, hemoglobin should dissociate with oxygen also. If they are going to bind irreversibly, let's say hemoglobin says, I don't want to leave oxygen, what will happen? Your cells will die of suffocation, your cells will not receive. So they bind in the reversible manner. Is that clear? Each one can carry maximum of 4 molecules. Already told you, yes or no? So each hemoglobin molecule carries 4 oxygen molecules. Now this is primarily related to the partial pressure of oxygen, because hemoglobin can bind with oxygen as well as CO2. How and why will it bind with oxygen? When will it bind with oxygen and not with CO2? It depends on the partial pressure of oxygen. Is that clear? So, and partial pressure of CO2, hydrogen ions concentration, temperature, these are also the factors which will determine whether hemoglobin is going to bind with oxygen or it is going to dissociate. Now we are going to study this with the help of a graph which is going to give us a sigmoid curve. Let's talk about this very important graph. Now see here. Okay, so here guys we have on this y-axis, what are we taking? We are taking percentage. We are taking percentage saturation of hemoglobin with oxygen. This is what we have taken along the y-axis. And along the x-axis, guys, we are going to take the partial pressure of oxygen in mmHg. So this is the x-axis. So first of all, you should know what are we taking along the x-axis and what are the parameters taken along the y-axis. Now, see. Yeah. Now see, when we plot this graph, we get this shape. This is S shape. We consider it S shape, and that is why we call it a sigmoid curve. We call it a sigmoid curve. This is a sigmoid curve, right? S shape. Now we call this graph as oxygen dissociation curve, ODC. Clear? So basically this graph tells us the story where and when oxygen is going to associate and dissociate with hemoglobin. So do we see here, that here the percentage saturation of hemoglobin with oxygen is 100% here. Now this percentage, 100% saturation, this can only occur at which pressure? Around, it is occurring at, it is occurring around this pressure. And this is nothing but 95 mm of mercury. Where do you see this pressure? Where do you see this 95 mm of mercury pressure? This pressure is found in the alveoli or in the lungs. So this is called 100% saturation. This occurs in alveoli, right? This occurs in alveoli. Clear? Now, what does S-shaped curve consisting of? It consists of three phases. First phase is, guys, known as the lag phase. Then we have the log phase or the exponential phase. And then we have this stationary or the plateau phase, also known as the constant phase. Now, what does it show? What does it indicate? So lag phase means first binding of first oxygen molecule is going to be slow. So this represents the binding of first oxygen molecule. Now when one oxygen molecule binds, the binding of the second and third molecule becomes very easy. Is that clear? So that is why it quickly binds, and this gives us exponential. Exponential means sudden rise, right? So this represents binding of second and third oxygen molecule. And then the fourth oxygen molecule also binds, and thereafter there is this plateau phase. Because one hemoglobin can carry a maximum of only 4 oxygen molecules. Please tell me these points are clear, right? And this is called cooperation. When one oxygen binds, the binding of second, third and fourth, it becomes easier, and then it is exhausted. It is all saturated. Please tell me these points are clear to all. Yes or no? Please tell me these points are clear. Yes, please. Diamond and all those students who are finding it difficult to find my Telegram channel, I'll get it linked with my video also. Also, you can always leave a comment and I will personally share it in the comment box. Don't worry. First, concentrate here. Now see, please understand, this curve is not static. This curve shows shifting on the right hand side as well as on the left hand side. How and when? Let's see. So when this curve is going to take the right hand shift, and when will it take the left hand shift? See, this is the left hand shift and this represents the right hand shift. What does this right hand shift and left hand shift indicate? See here. What does it indicate? First of all, the left hand shift indicates that, see, left hand shift is going to indicate that oxygen will bind to hemoglobin quickly. Why? Because there is high pressure of oxygen. So left hand shift will occur under these conditions when the partial pressure of oxygen is high but the partial pressure of carbon dioxide is low. If carbon dioxide is low, then the concentration of H positive ions is also going to be low. If H positive will be low, less H positive ions means more of pH. So high pH. Yes or no? High pH. So see, it is clearly written here which are the conditions which this curve is called dissociation curve, helps us to study the effect of factors like these. And now see here, in alveoli, what happens? It takes the left hand shift. So high pressure of oxygen, low partial pressure of CO2, lesser H positive ion concentration and lower temperature. And so less H positive means higher pH and then lower temperature. All these conditions will shift it towards the left hand side, and we see these conditions where in alveoli. So left hand shift indicates, it indicates oxygen hemoglobin association. It indicates, or you can say it favors oxygen hemoglobin association. Are these points clear? While the right hand shift occurs just the opposite, when partial pressure of oxygen is low but partial pressure of CO2 is high. When CO2 will be higher, more H positive ions will be formed. More H positive means lower pH or high acidity. Yes, and high temperature. So these five factors are going to shift the curve on the right hand side. And what does this indicate? It favors oxygen hemoglobin dissociation. It indicates dissociation. This favors association. Please tell me all these points are clear to you. So see, they all favor the formation of oxyhemoglobin. Yes or no? While in tissues, now talking about in tissues, low partial pressure of oxygen, high PO2, high H positive concentration and high temperature exist, and they favor the dissociation. Please tell me these points are clear. So this occurs in tissues while this occurs in the alveoli. Please tell me this ODC is super clear, and the factors which are responsible, guys, the factors which shift it towards right hand side and left hand side are clear. Please tell me these points are clear. Are these points clear? Yes? All right. Use a dark color pen. Okay, I'll use a dark color pen. Please tell me these points are clear. I'll take care of that color. All right. Now moving on to the next part, that is this very important line. See here. It says every 100 ml of oxygenated blood can deliver around 5 ml of oxygen to the tissues. Very important. Every 100 ml is going to transfer only 5 ml of the oxygen, while see here, should be written somewhere here. Okay, never mind. We'll just write it ourselves. See. So, every 100 ml of oxygen, see, let me put it clearly. Every 100 ml of blood carries, see, every 100 ml of blood carries 5 ml of oxygen and 4 ml of CO2. This is, of course, the deoxygenated blood we are talking about. This is, of course, the oxygenated blood we are talking about. Please tell me these points are super clear to you all. Are these points clear? Anyone with any doubt? Are these points clear? Yes, please. Are these points clear? Is that clear to everyone? All right. Now, moving on to the next, we have discussed this already. Now, transport of CO2. So, basically, see, carbon dioxide is carried by hemoglobin in the form of aminohemoglobin. We have discussed it already. And how much percentage? 20 to 25%. And when does it join? So, see, when it is written, the binding is related to the partial pressure of carbon dioxide. So, partial pressure of oxygen is a major factor that can affect this binding. See, if partial pressure of CO2 is high and partial pressure of oxygen is low, then more binding of CO2 will occur with hemoglobin. While if partial pressure of CO2 is low and this is high in alveoli, then dissociation will take place. This is something we have already discussed. Please tell me these points are clear. Now, how does this transport take place actually? Now, this is the reaction which is occurring in the RBCs. I'm talking about the transport of carbon dioxide. So this is your RBC, yes or no? This is your RBC. Now guys, and outside we have plasma. See, outside we have plasma. Okay. Now see, in your RBC, this carbon dioxide will enter and react with water in the presence of the fastest enzyme, that is carbonic anhydrase, and form carbonic acid. This again in the presence of the same enzyme will dissociate to form bicarbonate ions and H positive ions. Now this enzyme is present in plasma too. See, it's written carbonic anhydrase and minute quantities of the same is present in plasma. So it is present here. So the same reaction is occurring here. Same reaction is occurring here also. H positive and bicarbonate ions. But the difference is, since carbonic anhydrase is present in the lesser amount, what will happen? So bicarbonate ions formed here are in low concentration. They will be less, while bicarbonate ions formed here will be in the higher concentration. Yes or no? Yes. What will happen? One, within the RBCs, they are found in the higher concentration. In plasma, they are in the less concentration. Automatically, they will start moving out. So they will move from RBC into the plasma. What? Bicarbonate ions. Bicarbonate ions move out. That means efflux takes place. Yes. Now, in order to compensate, the chloride ions from plasma are going to move into the RBCs. Guys, this is known as the chloride shift. This is known as the chloride shift. And where does all this take place? Where will carbon dioxide enter into the RBCs? This is towards the tissue side, of course. This is towards the tissue side. So chloride shift will take place. Yes or no? Now bicarbonate ions which have moved outside, what is going to happen? These bicarbonate ions will react with sodium. These will react with sodium. Bicarbonate ions will react with sodium as well as potassium ions to form sodium bicarbonate, told you, and potassium bicarbonate. So that is why I said bicarbonate ions, 70% of CO2 is going to transport in this format. Yes or no? So this reaction is occurring at the tissue site where partial pressure of CO2 is high due to catabolism. Please tell me these points are clear. But now, when at the alveolar side partial pressure is low, the reaction will proceed in the opposite direction, leading to the formation of CO2. At the alveolar side, what will happen? These bicarbonate ions will move back, chloride will move out, bicarbonate and H positive will react back to form carbonic acid. They will again form carbon dioxide and water, and this carbon dioxide will move out in the alveoli. Please tell me these points are clear. Now there is a very important line. Every 100 ml of deoxygenated blood delivers 4 ml of CO2 at the alveoli. Please tell me these points are clear. Please tell me these points are clear. Yes, please. Yes, Ashwa. Chloride shift and hamburger shift, one and the same thing. Hamburger shift, also known as hamburger shift. So this occurs, this is all occurring towards the tissue side. Told you, this is all occurring towards the tissue side, and towards the alveolar side, there will be reverse of the reactions seen. Please tell me these points are super clear. And now coming on, now okay, before we move on, so guys, this itself is known as the Bohr's effect. What is Bohr's effect? Bohr's effect operates at the tissue level. And what does Bohr's effect say? Bohr's effect say when the partial pressure of CO2 increases, yes, so oxygen hemoglobin dissociation will occur, and this CO2 will bind with hemoglobin. Yes, this CO2 will also enter the RBCs and all the reactions will take place. Please tell me these points are clear. Dissociation will take place. And the reverse of this is Haldane's effect, which occurs when the partial pressure of oxygen is high at the alveolar level. Please tell me these points are clear. right? Are these points clear, everyone? Can we move on now? All right. Now coming on to the regulation of respiration. So which part of the brain or your central nervous system regulates the breathing, and whether your breathing rate will be high or whether you'll be breathing shallow or deep, right? So there are certain structures here and there are certain factors also. Now, first of all, in order to maintain and moderate the respiratory rhythm to suit the demands of the body, this is done by the neural system, right? Now, there is a specialized center in which region? Medulla oblongata, and we call it respiratory rhythm center. Is that clear? So this is responsible for this regulation. Second, it says another structure is found in pons varolii. Now this structure is called pneumotaxic center and it moderates the functions of respiratory rhythm center. Let me tell you clearly. See here. So one, it is medulla oblongata. Second, it is pons. Now in pons, guys, we have pneumotaxic center. What does pneumotaxic center do? Pneumotaxic center is actually going to decrease the duration of inspiration. This is a PYQ. So more the impulse from pneumotaxic center, so more the message that you get from pneumotaxic center, your inhalation, that duration will go on decreasing. So it's actually going to shallow your breathing, right? So we inhale, we exhale. So we'll inhale for the lesser time and we'll exhale more. That is what pneumotaxic center does. While medulla oblongata has respiratory rhythm center. Are these points clear? So you should know medulla has respiratory and PP. Pons have pneumotaxic center. Now the neural signal from this center, see it's written here, this center can reduce the duration of inspiration and thereby alter the respiratory rate. So lesser the inspiration, you are going to breathe more, right? Now, secondly, there is this chemosensitive area adjacent to the respiratory rhythm. This is highly sensitive to CO2 and hydrogen ions. Very important. So, more the CO2 in your blood or more the H positive ions, more is going to be your breathing rate. See, increase in these substances can activate the center. So, basically, more is the CO2 and H positive ions. So, more is the breathing rate. There is a line written out clearly in NCERT. See here, the line says the role of oxygen in regulation of respiratory rhythm is quite insignificant. So basically, if we don't have CO2 inside, we may not breathe. So it is the level of CO2 that is regulating. So increase in these substances can activate the center and in turn signal the breathing to make necessary adjustments in the respiratory that can be eliminated. So receptors for these chemosensitive areas. So the receptors, see here, the receptors are found in two places. One is aortic arch and then the carotid artery. So the aortic arch is the one that comes out of the heart and carotid artery is the vessel that carries blood to the brain. So these two blood vessels have chemoreceptors for CO2 and so these are the not just any receptors, these are the chemoreceptors. These are the chemoreceptors for CO2 and H positive ions. Is that clear? And they will send signals to rhythm centers for remedial actions. The role of oxygen is quite insignificant. Please tell me that these points are clear to everyone. Please tell me these points are clear to everyone. Yes, please. Clear? So you have learned. Okay. Now before we move on, we have to talk about the disorders. Let's discuss the disorders here of respiration. What are the different disorders? First of all, we have asthma, we have bronchitis, then we have emphysema, and we have occupational lung disorders. Occupational lung disorders. Right? Let's start from the first, that is asthma. Now, both asthma and bronchitis, these are basically the allergic reactions. These are the allergic reactions. Number one, this as well as this. Itis, inflammation. What is going to happen in these cases? There will be difficulty in breathing, and particularly in asthma, there is a characteristic sound which is called wheezing sound produced during exhalation. So while you're breathing out, there is this whistle-like sound produced, wheezing sound produced during exhalation, right? It is actually during the contraction of terminal bronchioles. Now, in order to open up these terminal bronchioles, we need to take, what do we take for these asthma and bronchitis? We take bronchodilators, right? Those inhalers, they contain these. Is that clear? That is asthma. Then, talking about emphysema, what happens in emphysema? There is destruction of alveolar walls. Destruction of alveolar walls. Or you can say damage, degradation of alveolar walls. Now, why does it happen? The main cause is cigarette smoking. The main cause of emphysema is smoking, and we must know that it is a non-irreversible change. Once the damage has happened, it is going to be permanent. Please tell me these points are clear. And then, as the name says, occupational lung disorder. What is occupational lung disorder? It is a lung disorder because of a certain occupation. Of course, not the white-collar jobs. So there are certain occupations where the workers, they deal with fine particles, for example, cement, cotton fibers. I'm not talking about the people who sit in their cabins. I'm not talking about the white-collar jobs. I'm talking about the labor class who's actually dealing with, who's handling these microparticles of cement, cotton, then glass, asbestos, okay, etc. All these are the microparticles. They settle down. These enter the lungs with the along with your breath. Okay, so microparticles, they enter the lungs and they cause proliferation of fibrous tissue. Proliferation of fibrous tissue. Now, too much proliferation of fibrous tissue is going to decrease the surface area. So the space that has to be empty for the gaseous exchange, it is all proliferated with the fibrous tissue. So this proliferation of fibrous tissue leads to decrease in the surface area. Decrease in the surface area, and hence the disease, and it is called occupational lung disease. So according to the Factory Act, guys, Factory Act is meant for the safety of the workers. It says that workers should wear masks. Workers should wear masks. Please tell me these points are clear to everyone, and these are the disorders of this chapter. Please tell me, are these points clear? Yes, please. Are these points clear to everyone? All right, guys. Moving on now to the next chapter of human physiology. So that is body fluids and circulation. So what does this say here in this topic? See. So also the waste. See, you have learned that all living beings have been provided with nutrients, oxygen and other essential substances. The waste or the harmful substances produced have to be removed continuously, and therefore essential to have efficient mechanism for the movement of these substances from cells and to the cells. And different groups of animals have evolved different methods. Now, first of all, in simple organisms like sponges, coelentrates, how do these circulate their substances? They circulate the water from the surrounding through the body cavities to facilitate through the body cavities. Let me know the type of cavity present in sponges. Quickly let me know. We have read that yesterday. What is the name of the cavity? Not the body cavity, but the cavity which is present in porifera. Do you remember? The washing machine style? Yes. Canal system, spongocele, very good. the spongocoel or the paragastric cavity. And similarly, coelentrates have coelentron, the gastrovascular cavity, right, which helps in circulating the water through their body cavities to facilitate the cells to exchange the substances. Similarly, more complex organisms like have special fluids within the body to transport the materials, right? More complex have special fluids. For example, blood is the most common used body fluid in higher organisms for this purpose. Another fluid is lymph, which also helps in the transport of certain substances. We are going to talk about this in detail in this chapter. First of all, let's start with blood. Now, blood, guys, represents the specialized connective tissue. It represents a special connective tissue, yes or no? And it constitutes the fluid matrix, plasma, and the formed elements. So these are the two components of blood. One is the liquid part. Liquid part is known as plasma, while the cellular part, which is called formed elements. And all the formed elements consist of cells like RBCs, WBCs, and platelets, right? Plasma is the liquid part. It constitutes 55 to 60% of the total, while, see, it is written 55%, and this constitutes the rest of the 45%. Is it clear? Now, the plasma, if I will centrifuge blood, you will see all the formed elements to be forming different layers here. Okay, they form the different layers, while there is yellowish colored fluid which will be floating on the top. Yellowish colored fluid floating on the top. Is that clear? So this yellowish fluid is called plasma, while rest of them, they are the cells. So like this. Now let's talk more about plasma. It is straw colored, not exactly yellow, yellowish, viscous fluid for constituting 55%. And now, what is the composition of plasma? What does plasma has? It has 90 to 92% water. It is water. Proteins, they constitute 6 to 8%. This was a PYQ in the form of match the following. Now, what type of proteins are present? See here, the proteins present are albumins, globulins, and fibrinogen. Now, this fibrinogen is the largest of all the proteins. Albumins are the smallest. What are the functions? Let's see. Fibrinogen plays important role in clotting mechanism, clotting of blood as and when needed. Albumins, they maintain the osmotic balance of blood. Osmotic balance means how much water will be present in blood. So that is determined by albumins. And then we have globulins. Globulins are going to these are globular proteins, and they're going to fight against the infection. Fight the infections. Please tell me these 3 points are clear. So these are the 3 major proteins. Apart from these 3 proteins, we have different other proteins as well. Is that clear? Moving on to now, see, it is already written fibrinogen, important role in clotting or coagulation. This globulins in the defense mechanism, so basically antibodies, and albumins, they are the smallest and help in maintaining the osmotic pressure. Now, other than this, our plasma has minerals like these, then glucose, all the nutrients are present in the plasma. They transport these things. Now, factors for coagulation, the other factors, the clotting factors are also found in plasma. Is it clear? But they are all found in the inactive form. If they are already in the active form, your blood will clot. Now, that can be a serious matter. Now, very important line. So, plasma without clotting factors is known as serum. So, if you subtract the clotting factors from the plasma, that will form the serum. Please tell me these points are clear. Please tell me these points are clear. What is not visible here? Okay, red is not visible on white. I'll take care of this. Is that clear? All right, guys. All right, noted. Okay, Now, moving on to the formed elements. What are formed elements? Formed elements consist of these cells, erythrocytes, RBCs, leukocytes, your WBCs, and platelets. Together are known as formed elements. How much to, what is the percentage of formed elements in blood? That is 45%. Now, talking about the RBCs, RBCs are the most abundant. See here, RBCs are the most abundant of all. How much are they present? On average 5 million to 5.5 million in per millimeter cube of blood. You have to remember the value. Where are they formed? Formed in the red bone marrow. So red bone marrow is the place, this is the place of erythropoiesis. This is erythropoiesis. This is the site. This is where blood formation or RBC formation takes place, site of erythropoiesis. Poiesis means formation. Now, RBCs in mammals and especially in humans, they are devoid. In most of the mammals, they're devoid of nucleus. Why are they devoid of nucleus? Just to increase the surface area. The shape is also biconcave. Again, this shape has maximum surface area. So, the surface area is being enhanced to accommodate more and more of hemoglobin, and we have already seen the importance of hemoglobin in the previous chapter. It is red colored iron containing complex protein. Now the structure of hemoglobin is quaternary, and we have read that in the chapter of biomolecules yesterday. There are 2 alpha and 2 beta, right? Hence the color and the name of the cells. The healthy human being has 12 to 16 grams of hemoglobin. So this is important. How much hemoglobin? 12 to 16 grams per 100 ml of blood. You also have to remember 12 to 16 grams in how much blood? 100 ml of blood. Now what is the function of hemoglobin? We have already studied that it helps in the gaseous exchange, transport of respiratory gases. So on average, the lifespan is 120 days, after which it is going to be destroyed in spleen. So that is why spleen is called the graveyard of RBCs. But in case of emergency, it is also the reservoir of RBCs as well, but only in case of emergency. It is going to release this emergency blood bank. So only during the emergency, it is going to release the RBCs. Otherwise, normally it acts as a graveyard. Please tell me these points are clear. Yes, guys. Hi, Vishwa, you're late. Don't worry. We have started with the second chapter of human physiology, and you can cover up the previous one later. So let's go with the flow. Is that clear, everyone? Graveyard, yes. Ma'am, what is spleen? Hayota, spleen is present. So say this is your stomach, and so this side I have my stomach, that is on the left hand side, and behind the stomach we have spleen. Behind the stomach we have this structure called spleen. Okay. Now, and spleen is the largest secondary lymphoid organ also. We'll talk about spleen more during the chapter of human health and diseases. Okay, are these points clear? Now talking about leukocytes here, leukocytes also, leuko means white. So leukocytes are the WBCs. They are colorless. Why? Because they lack hemoglobin and they are well nucleated and relatively less in number. How many less? They are only 6,000 to 8,000 in number per millimeter cube. And they are generally short-lived because they are soldiers of the body, so they might die. They are protecting us all the time. So there are two main categories, guys, of WBCs. One is granulocytes and second is agranulocytes. Now neutrophils, eosinophils, basophils, these are the types of granulocytes because they have granules, right? While these lymphocytes and monocytes, these are agranulocytes because they don't have granules. So as the name says, granule present, agranulocytes, granules absent. Out of these guys, neutrophils are the most abundant. How much? You must remember the percentage. This year, they have given a question also based on the percentage, and you've been asked, and they've asked that very cleverly. You are supposed to calculate. So you must know the percentage here. Neutrophils 60 to 65, right? Basophils, they have least 0.5 to 1%. Then monocytes are just 6 to 8%. And then we have basophils, okay, we have eosinophils which constitute 2 to 3%. Yes or no? Yes or no, guys? Right? So what are the functions? What are the different functions? Now let's see the function of each. Neutrophils are basically phagocytic. See, neutrophils and monocytes, they are both phagocytic, right? Talking about basophils are secretory. Written, they secrete. What does it secrete? It secretes number 1 histamine, number 2 serotonin, and number 3 heparin. So what is the function of histamine? It is inflammatory. It causes inflammation. It is inflammatory. Serotonin, it is antagonistic to that, anti-inflammatory. And heparin is a clotting agent, anticoagulant. It is an anti-clotting agent. So your blood doesn't clot inside because of the heparin present. Please tell me all these points are clear. Please tell me all these points are clear. Yes? Okay. So what is the function of basophils? Yes. What is the function of basophils? So basophils are going to secrete histamine, serotonin, and heparin. Function of eosinophils. They have special role during, they resist the infections, and also they have important role during allergies. So this is the main function of eosinophils. So whenever there is allergy in your body, the level of eosinophils increase. So these are the structures, this is the figure which is given in NCERT and indicates the different types of blood cells. Let me tell you more about eosinophil. So eosinophil, I'm going to draw here. Eosinophil has bilobed nucleus. See here. So eosinophil, this is eosinophil. It has bilobed nucleus. Is it clear? So nucleus is one, but it has two lobes, 1 and 2. Clear? Then basophils have roughly S-shaped nucleus. So if you have to identify on the basis of nucleus, so it has S-shaped nucleus. Neutrophils have multi-lobed nucleus, and the shape is not constant. So multi-lobed nucleus. Then monocyte is the largest of all, and it has a kidney-shaped nucleus. Reniform or kidney-shaped nucleus. And then lymphocytes, be it T or B lymphocytes, are the smallest, and maximum space is occupied by nucleus. So there is hardly any space left for the cytoplasm. So do you see that monocyte? This is monocyte. Let's put the tag. These are monocytes. This is neutrophil, this is basophil, and this is eosinophil. These are lymphocytes. Do you see lymphocytes are the smallest? Monocytes are the largest. Neutrophils are the maximum, and basophils are the least. So how do you remember them in the decreasing order? It is null map. It is null map. Can you remember the sequence in the decreasing order? Yes or no? Please tell me. Can you remember that now? Can you match the following? Other than this, guys, why do we call eosinophil? By the way, there is another name for eosinophil, which is acidophil, also known as acidophil, because they have acidic granules. The basophils are called basophils because they have basic granules. They'll appear blue on staining. Neutrophils will have both acidic and basic granules, while these are two agranulocytes. They will not have granules. So these three are the granulocytes. Please tell me these points are clear to everyone. Yes, please. Please tell me these points are clear to everyone. Yes or no? Is that clear? Is that clear? Now, neutrophils can also be known as PMNL, that is polymorphonuclear leukocytes. This is another name, AKA. Please tell me these points are clear to all. Alternate names done, Afan? Yes. All right. Now moving on to next. So see, we have already discussed that lymphocytes constitute 20 to 25%. There are two types, B or T. We'll read more about that in health and diseases. B are because they are differentiated in the bone marrow, and T because they are differentiated in the thymus. They constitute your AMIS, antibody mediated system, and T are responsible for cell mediated. The details will be discussed in the other chapter. Now talking about platelets. Okay, platelets also known as thrombocytes. Now platelets are not actually real cells, they are the cell fragments. They are very sticky, and the mother cell of platelets, they are produced from megakaryocyte. Megakaryo because the original mother cell of platelet has a large nucleus, megakaryocytes. They formed in the bone marrow, of course. What is the value or the number of platelets? What is the platelet count? That is 1.5 lakh to 3.5 lakh platelets per millimeter cube. This is the normal amount. And we know what happens when platelets go down. They are released with, so platelets play a very important role in coagulation or clotting mechanism. So if the number of platelets fall, the clotting mechanism gets disturbed. Now it can lead to internal bleeding and hence death also. So it leads to clotting disorders and lead to excessive loss of blood from the body if there is reduction in the number. Please tell me these points are clear. And you also know that the number of platelets decrease markedly during dengue and chikungunya. Yes? Yes, guys? Please understand. Let me know. All right, guys. All right. Now, moving on to the blood groups. Now, blood groups, basically, the blood can be grouped on the basis of some antigens, some special glycoproteins present on the RBC surface. So, if this is the RBC, there are many antigens present. There are some antigens present on the surface. So, these antigens help us in grouping the blood. Okay, now depending upon the antigens, we have different types of blood groups. See, we are going to follow two types of blood group system. One is RH system and ABO system, and these two systems are sufficient for the successful blood transfusion. Is that clear? Is that clear? Let's talk about the ABO blood group system here. Say, ABO grouping, it depends on the presence or absence of two surface antigens. That's what I've told you, which induce the immune response. Is that clear? Now, distribution of antigens, and accordingly we have 4 blood groups, and we have to consider these blood groups for successful blood transfusion, otherwise it can be deadly. Now see here, this table explains everything and the donor compatibility. So I told you, so the blood group depends upon the antigen. So the antigen on the RBC, that is the same blood group. So if you have A type of antigen, it is A type of blood group. B antigen, B type of blood group. If you have both the antigens, A and B, then the blood group will be AB. Is that clear? So RBC would be A and B. It has A and B. While in here, it will have only B. In here, it will only have A. What about O? O is actually 0. O basically means 0. There is no antigen. There is no antigen. So it is naked like that. Okay. Now because there is no antigen, that is why it makes it universal donor. Why is O a universal donor? Because there are no antigens present, because these antigens, see it is written, these antigens can induce the immune response. Yes or no? So because there are no antigens, so it can be donated to anyone. There will be no immune response generated. Second point to be noticed, is the antibodies in plasma. See, whatever antigen you have on RBC, the antibodies are present reverse to that. So if we have A antigen, the antibodies would be anti-B. See, antibodies are meant for protecting you. So if I have A antigen, my body is going to protect me against B, right? So I will have anti-B. Similarly, if I have RBC with B antigen, my body is going to protect me against A, so anti-A. Now, since there are both antigens present in case of AB, so body recognizes both. So there will be no immune response generated if any enters. So there will be no antibodies. So since there are no antibodies in case of AB, therefore they can take blood from anyone and hence they are the universal recipients. Why? Because there are no antibodies. So remember, O is a universal donor because of no antigen. AB is a universal recipient because of no antibody. Are you getting that? While your O blood group individuals have both anti-A and anti-B. Yes or no? Yes or no? So I think this is super clear. So who all can donate blood? Donors group, who can donate blood to A? One is of course A, and then we have this social server, that is O. Then B can take blood from B as well as O. AB can take blood from all, the universal recipient, right? And then O can give blood to all. That is why it is a universal donor. Yes? Please tell me these points are clear. Yes or no? Yes, guys. Quickly let me know. Coming on to the positive negative aspect also. Wait a second. Wait a second, guys. Now see here. Other than ABO, there is another antigen which is called RH antigen. RH antigen. RH stands for rhesus. Now this antigen was first discovered in rhesus monkey, where it was first discovered. Is that clear? Where it was first discovered, in the rhesus monkey, and we carry that name RH. Now, the RBCs which have this RH factor will be known as RH positive, while those without RH will be known as RH negative. So, apart from A and B, some individuals will have RH also. So, they can be A and RH positive. We simply call it A positive. They can be B and B, RH positive, so B positive. If they don't have RH and just A, we'll call it A negative. Please tell me that this point is clear. This point is clear, right? Now, the main point comes when there is marriage between two different RH individuals and the pregnancy. Now, first of all, understand what is going to happen. So, let's say the father and the mother and the baby. So, if the father is positive, mother is positive, the baby will be positive, right? If father is negative, baby will be negative. Is that clear? Now, if father is negative, mother is positive, one is positive, baby will be positive. I am talking about RH. So, this is RH, okay? Now, if father is positive, mother is negative, again, one of the parents is positive, baby will be positive. Yes or no? Now, let's keep the father aside. Now, the baby is going to grow inside the mother. So, let's see if the RH factor of mother and baby, they match or not. So, mother is positive, baby positive, fine, no problem. Mother negative, baby negative, no problem. Mother positive, baby positive, no problem at all. The problem arises when the father is RH positive, mother is RH negative, and the baby becomes RH positive. Now, this is the case which leads to RH incompatibility between mother and the baby. First question is here itself. It is the RH incompatibility between mother and the baby. Is that clear? So, what happens now? The problem arises. Now, see, let's say this is the mother and this is the fetus. Is that clear? The mother and the fetus. Please tell me, the points are clear so far? Now, there is this placenta. I'm going to draw this as placenta. This is just a pictorial representation, guys. Okay. Now what happens during the first pregnancy? See, during the first pregnancy, during pregnancy, be it first or second or any, during pregnancy, the blood of the mother and the baby does not come in contact directly. There is just exchange of substances from maternal blood to fetal blood and fetal blood to maternal blood. The real, like the blood actually doesn't come in contact. It might come in contact during the delivery. Is that clear? So, the first pregnancy is absolutely normal. Absolutely normal. Now, what happens if during the delivery, remember, the fetal blood comes in contact with the maternal blood, that's where the whole issue begins. Then what will happen? Now, during the delivery, if fetal blood comes in contact with the maternal blood, mother's body starts making antibodies against Rh factor. Now, let me bring to your kind notice, like unlike in case of ABO system, you are already born with the antibodies. So, if you are A, you are born with anti-B. If you are B, you are born with anti-B. But that's not the case with Rh. If you are born Rh negative, you will not have antibody against it since birth. It will develop only and when you come in contact with the Rh factor, like in this case. Now, mother's body, mother is negative, right? So, see here. So, this is the mother. Mother is Rh negative, but because of the Rh positive husband, this fetus is Rh positive, right? Now, if during the delivery, this blood comes in contact with the mother will start making antibodies against the Rh factor. Now, which antibodies will she make? She will make the largest antibody, that is IgM. Now, these antibodies will do nothing in the first pregnancy because the baby has been delivered already. But during the subsequent, now see, during the subsequent pregnancies, that is next pregnancy, every next pregnancy is going to be an issue here. So, during the subsequent pregnancies, what will happen? Now, every time this Rh comes in contact with the mother's body, now mother's body is going to produce, what? The antibodies. Mother is going to produce IgG this time. Remember, IgG is produced during the secondary immune response. So, this IgG is going to enter the fetal blood or the fetus. IgG will be produced and this IgG will enter the fetus body. This IgG is the only cross placental. Do you remember? This can cross placenta. Cross placental or crosses placenta, and IgG destroys the fetal RBCs. Please tell me these points are clear. Now, when the RBCs of the fetus are destroyed in very large amount, it will lead to this it destroys the fetal RBCs. Now, this is going to lead to severe anemia, yes, as well as severe jaundice because of degradation of hemoglobin, also severe jaundice. And both these conditions are enough to kill the baby. So, that is why subsequent pregnancies will result in stillbirth. That means dead baby will be born, and hence we call this situation as hemolytic disease of newborn. Hemolytic disease, blood lysis, hemolytic disease of newborn, HDN. Also, it is known as, or we call it, erythroblastosis fetalis. Please tell me these points are clear. Erythroblastosis fetalis. Yes, guys. Please tell me these points are super clear to all. Yes, guys. Are these points clear? Quickly give me a thumbs up. Everybody, all those of you who have been focusing and not just chatting, quickly give me a thumbs up. Let's have a thumbs up break. Yes, please. Let's have a thumbs up break. Have a sip of water if you're feeling sleepy. What happens in case of twins? Depends on twins. Doesn't matter. Twins will also die. Monozygotic twins. Yes. Is that clear? Okay. Now, talking about, see, so how do we, so, but these days it is not a big issue. Just that the right type of blood test should take place, should be done, and now this situation can be prevented. See, this is avoided by administering the anti-RH antibodies to the mother immediately after the delivery of the first child in this case. This line is important. Immediately after the delivery. Is that clear? So these anti-RH antibodies are going to protect the mother from producing these antibodies, and they're going to protect from the subsequent stillbirths. Is that clear? Is that clear? All right. So we have discussed about the blood groups, not talking about the blood coagulation, that is clotting mechanism. Now blood should clot when there is a bleeding. Okay, otherwise it can lead to excessive bleeding, which can be fatal as well. Now, see, usually the blood stops flowing after some time. Do you know why? Because it exhibits coagulation. There is a proper mechanism, and we have seen that reddish brown scum formed at the site of cut. What is that? It is a clot or a coagulum. So where is it formed? It is formed by a network of threads called fibrins, in which dead and damaged formed elements are also trapped. Now, fibrins are formed by the conversion of inactive fibrinogen. Now, inactive fibrinogen, they are activated by the enzyme thrombin. Thrombins in turn are formed from another inactive, that is prothrombin. Now, prothrombin becomes thrombin because of thrombokinase. This is required for the above reaction. All these are going to occur one by one. They are a series of linked reactions, so we call them cascade reactions. So there are a number of factors involved, but all these are found in the, so number of factors present in plasma in the inactivate stage. So only the injury or the trauma stimulates the platelets to release certain factors which initiate coagulation. A very important line, calcium ions play important role in clotting. Now we have not understood, or we have understood. So see, what NCERT has written is just the opposite. I am going to write it clearly here. So basically what happens whenever there is a tissue injury, platelets are very delicate cells. Platelets will burst and they release, this enzyme called thrombokinase. The platelets release, I'll just write down here, platelets release. So whenever there is tissue injury, platelets are going to release thrombokinase. As the name says kinase, it is going to be an enzyme, of course. So who releases platelets? Sorry, thrombokinase, it is released by platelets. Now this thrombokinase is going to convert prothrombin into thrombin. Yes. Now prothrombin, as we know, is inactive, while this is the active form. Right? So once the prothrombin is changed into thrombin, once this thrombin is formed, this thrombin is going to activate fibrinogen and form fibrins. Again, now fibrinogen is a monomer basically. It is a single thread. While, and we have read about this fibrinogen, they're found in the blood, right? These are the largest proteins and help in clotting. So that's how they help. And they will polymerize to form polymer to form fibrin. So fibrin is basically this network. It is going to form this network. So each thread is a fibrinogen and it is going to polymerize to form this fibrin network, right? and it is written in which dead and damaged elements of the cells are trapped. So these are the dead cells trapped in. So dead cells are trapped, right? And together this thing is known as a clot or a coagulum. Are these points clear? So do you see the hierarchy? Do you see the series of steps that are going to take place? So one step will be proceeded by, it is just a matter of platelets releasing the thrombokinase, and rest will automatically follow. See here. Now prothrombin and fibrinogen and all these are all clotting factors. These are all clotting factors, and they have been produced by your liver. So liver has produced this, and liver has produced fibrinogen and put it in your blood. Is that clear? And you need vitamin K to produce prothrombin. So that's where the role of vitamin K comes in, and calcium ions are going to help in this process. Please understand and please tell me that this process is super clear. Now, do you understand how things have taken place for coagulation? Please tell me, guys. Factor 4 hemophilia. Yes. Now see, overall guys, there are 13 factors. Numerically, there are 13 factors responsible for blood clotting, but we don't have all those 13 factors in our syllabus now. Some of the factors are produced by your DNA, by your genes. So yes, there is a genetic control also in regulating the blood clotting mechanism. Is that clear? For example, hemophilia that you talk about in genetics. So in hemophilia, what happens? There is lack of some clotting factor, right? And then excessive bleeding occurs. Okay. Anyways, now talking about the lymph, which is also known as the tissue fluid. Now this is also another specialized connective tissue. This is also a specialized connective tissue, just like your blood. Specialized connective tissue. What is the difference? So some water along with some water soluble substances move out. So some water along with water soluble substances move out in the spaces between the cells, leaving the larger proteins and most of the formed elements in the blood vessel. This is known as lymph. So basically, lymph is formed from the blood and goes back into the blood. So from the blood, we are going to take out plasma, that is lot of water, some soluble substances, and the name lymph comes from lymphocytes, so we'll have some WBCs also here, and that's what blood minus all these things, and that's what is going to form lymph. Please tell me these points are clear. Please tell me these points are clear, right? So the fluid released outside is called interstitial fluid or tissue fluid, or we call it the lymph. It is the same composition. It helps in the exchange of nutrients, gases between blood and the cells as occurs through this fluid. The elaborate mechanism, we have different lymphatic, we have a proper lymphatic system which collects the fluid, drains it back into the major veins. So basically, this blood is going to, this all comes out from the blood capillaries. So blood is going to release all these things and they are going to form the lymph. They are going to form the lymph. Is that clear? And it will be again drained back into the blood through veins. Is this clear? Is this clear? So lymph is colorless because there is no hemoglobin. It has specialized lymphocytes, told you already, which are responsible for the immune response. It is also the important carrier for nutrients and hormones. Very important function. Fats are absorbed through lymph in the form of lacteals present in the interstitial villi. Now I'll explain this part. So basically what happens here, so your intestine has these finger-like processes known as villi. These are intestinal villi. These villi are going to increase the surface area, right? Now beneath these we have our blood vessels. We have blood capillaries, right? So most of the things will be directly absorbed into the blood, but not the fats, not the fats. So there is this lymph capillary also. So these lymph capillaries under the intestinal villi, these are known as lacteals. These are known as lacteals. So fat, being a big molecule, is first put into the lacteal, and then through this whole lymph circulation, when the lymph will move back into the blood, fat molecules will be added into the blood. Is that clear? So they play a very important role in fat absorption. So lacteals are the lymph vessels or lymphatic capillaries under intestinal villi, under villi, an important role in fat absorption. Are these points clear? Please tell me. Please tell me these points are super clear to all. Is that clear to all? Anyone with any doubt? Hemophilia is X-linked. Yes, it is caused. We will talk about hemophilia more during that chapter. Wait for genetics. If I'll start the genetics here, the human physiology will be gone. So we'll take things one by one. Is that clear? We'll take things one by one. Now, after having discussed the fluids of circulatory system, we are going to talk about the circulatory pathway. Now see here, the circulatory pathways can be two. We have already discussed about this in animal kingdom. I've told you what is open circulatory system and what is closed circulatory system. So open circulatory system is found in all except two. So the two which have closed circulatory system is annelid and chordata. Who can tell me in the chat that which chordata will have open circulatory system? Rest all of them will have open circulatory system except Porifera, Coelenterata, Ctenophora, Platyhelminthes. They don't have circulatory system, right? And even Esculmenthes. So it will be found in Arthropoda, Mollusca, Echino, right? Hemi. They all have open circulatory system. Please tell me which chordata will have open circulatory system. Yes, guys. Cephalochordata, urochordata. Are you sure? Are you sure? Yes, guys. Cephalochordata, urochordata. They have open circulatory system. Primata, primula. Oh, Arjun, please focus. Yes, guys. Uro and cephalochordata. Please be sure. Hemichordata is not a chordata, Vishwa. I am asking about the chordate which has open circulatory system. Hemi is already a non-chordate. Now see, what is the difference? We have already discussed. In open circulatory system, guys, the blood vessels, large vessels, they will release the blood in the open spaces or the cavities called sinuses. Why? Because the capillary system is absent. Why? Because capillaries are absent. While in case of closed circulatory system, the heart is going to pump blood and blood will be only passing through the closed network of blood vessels. This is obviously more advantageous because you can regulate this blood. In case of open circulatory system, guys, blood cannot be regulated. All the cells are bathing in blood. So every cell and every tissue will receive almost the same amount of blood. There will be very less blood pressure. You cannot regulate that, okay, as per need, if let's say your muscles require more blood, then that blood can only be sent towards the muscles if blood is flowing through capillaries. If it is only flowing through special channels. So that can only happen during the closed circulatory system. So here capillaries are present. So organs are not bathing in blood, which is the case with open circulatory system. Please tell me these points are clear. Right? Now, all vertebrates, we have seen it yesterday that some of the non-chordates do have heart, but not all of them does. But all vertebrates will have a muscular heart, muscular chambered heart. Like fishes, for example, they have two-chambered heart. We have discussed it with atrium and ventricular. We have discussed it in detail. Amphibians and reptiles, except crocodiles and alligators, they have three-chambered heart with two atria and single ventricle. Whereas crocodiles, birds and mammals, they have four-chambered heart, right? Two atria, 2 ventricles. Now in case of fishes, we have seen fishes will pump only the deoxygenated blood. We have seen this in animal kingdom in detail. Yes, fishes will only, the blood of the, the heart of the fishes will only pump the deoxygenated blood. That is why, guys, we say that fishes have a venous heart. They have a venous heart because they only pump deoxygenated blood. So the deoxygenated blood is pumped by the gills and to the gills, and gills are going to supply it to the body. Is that clear? Now there is single circulation. We have also seen the blood enters the heart only once in case of fishes. Those of you who have not attended yesterday's lecture, who are finding this difficult, please watch the lecture. You will understand single circulation in detail. Then talking about amphibians and reptiles, guys, left atrium receives the oxygenated blood, gills and lungs, and the right atrium gets the deoxygenated blood from the other body parts, and they get the mixed up blood in the single ventricle, which pumps out mixed blood, and hence there is incomplete double circulation. Why is it incomplete? Because they are pumping out the mixed blood. While if we talk about birds and mammals, the oxygenated and deoxygenated blood are passed on to the ventricles, and there is no mixing, without any mixing up. So there is a separate circulatory pathway, hence we call it double circulation, and that too complete double circulation. We call this circulation as complete double circulation because there is no mixing up of the bloods. Please tell me these points are clear. Please tell me these points are clear. Yes or no? Now moving on to the human circulatory system. Now specifically about humans, see here. Specifically about humans. Now human circulatory system consists of blood vascular system, right? Why? Because there is blood as a main fluid. Then it has a heart. It has a closed network of branching blood vessels, and of course blood. So these three things constitute the human circulatory system, and it is also known as blood vascular system. It consists of blood, it consists of vessels, and the pumping organ called heart. Is that clear? Now let's talk about heart in detail now. Now heart is a mesodermally derived organ. It is mesodermal in origin. Where is heart present? Right in the center between the two lungs, right in the thoracic cavity. In the thoracic cavity between the two lungs, slightly tilted to the left. That is the only reason why you feel your heart towards the left, right? Now the size of the clenched fist. So when you have clenched your fist, but actually it is this is your heart. Is that clear? So it is the size of the clenched fist. There's a double wall membrane cover known as pericardium. So this is going to be the protective covering, and it has pericardial fluid. There are 4 chambers, guys. We know about that. 2 atria and 2 ventricles. Now there is a muscular wall between them that separates it. Interatrial septum separating right and left atria, and interventricular septum. Now this one is thin while this is thick, which separates left and right ventricles. Right on the same side there is a thick fibrous tissue called atrioventricular septum also. Now they are provided with the opening for the 2 chambers, and the opening between the right and left is guarded by valves. Now these valves are the muscular flaps, and we call this as tricuspid and bicuspid. Tricuspid is on the left side, right side, and bicuspid, also known as mitral valve, between the left atria and left ventricle. Is this point clear? Now let's see the structure first ourselves, and then we will proceed. Let me draw the structure for you so that we can understand it in a better way. See here. So this is your roughly triangular heart. This is your roughly triangular heart. Okay. Now this side is the apex. This is the apex. This is the base. Okay. Now your heart has 4 chambers. Heart has 4 chambers. Yes? Like this. So this side is your right auricle or right atria, and this is your right ventricle. Yes? While this is your left auricle and this being your left ventricle. Clear to all? Now what did it say? First of all, there is a protective covering. There is a protective covering, and again this protective covering is double layered. We call this protective covering as pericardium. So there is outer pericardium, there is inner pericardium. These are two pericardial layers, parietal and visceral, pericardium. And there is fluid present in between. There is a pericardial fluid. Is that clear? Now, it is clearly written that there is, okay, wait. Yes, please. Now, it is clearly written, there is a thin membrane present between the two atria and a thick membrane that separates the two ventricles. So, this membrane which separates the right and the left atria, this one is called the interatrial septum, and this is thin as written in NCERT clearly, while this between the two ventricles is known as interventricular septum. Yes or no? Yes or no? While the interventricular septum is thick. Is that clear? Now this portion between the two atria and ventricles, this is known as atrioventricular septum. Yes or no? Is the diagram coinciding with your theory? And this is thick and fibrous. Is that clear? So septum means which separates. Okay. Now also it is said that the right atria and right ventricle, they are connected through these flap-like structures. They are muscular flap-like structures known as valves. Those on the right side are the tricuspid valves because there are 3 flaps, and those on the left side is the bicuspid valves because there are 2 cusps. Other name is mitral valve. Please tell me these points are clear. So you should be very clear. On the right side we have tri, on the left side we have mitral. Is that clear? Is that clear to all? Yes or no? Okay. So do you see that the walls are not very smooth? The walls of the atria are not smooth. The walls of the ventricles are not smooth either. Now, these ridges present in the atrial and the ventricular walls, these ridges are going to increase the surface area. These are the ridges. Is it clear? Now, these ridges in ventricles are known as columnae carneae. Known as columnae carneae. Yes or no? Okay, these are known as columnae carneae. Now, on these columnae carneae, guys, are present some special muscles. These muscles are known as papillary muscles. These are known as papillary muscles. And through these papillary muscles are present some special thread-like structures which are going to hold these valves. Is that clear? So these thread-like structures which hold these valves are called, these structures are known as chordae tendineae. They are collagenous threads. Chordae tendineae. What are they made up of? Collagen, the most abundant protein, very strong. Is that clear? Please tell me these points are super clear. Right? And now, since our heart is auto-excitable, it is myogenic, so there is that structure where the beat, where the impulse originates. Now that structure is present here in the right auricle. This is known as SAN. So where is SAN? It is in the right corner of the right auricle. Please focus on the position of each and every structure. While there is AVN, auricular ventricular node, on the lower left corner of right auricle. So both these structures are found within the right auricle only or right atria only. Are these points clear? Please tell me. Are these points clear, everyone? The function of pericardial fluid is exactly the same, which is same as that of pleural fluid. Prevents the friction, prevents desiccation, right? So yes, it is the same function. Please tell me these points are clear. So guys, this is the structure of the heart. Is it clear? So what is the function of valves? Valves, wherever they are present, they always prevent, they allow the flow of blood only in one direction. So basically, they prevent any backward flow. So once the blood flows from right auricle to right ventricle, what? It should not be able to go back. Similarly, the blood which flows from left auricle to left ventricle, it should not be able to go back, right? So that is why once the valves close, these chordae tendineae, do you see here? Chordae tendineae are going to hold these valves from below so that it does not open into the opposite direction. So chordae tendineae are very, very important. They also help the valves from preventing them to open in the backward direction. Is that clear to all? Is that clear? I have not drawn any blood vessel here yet. Now the entire heart is made up of the cardiac muscles. So there are three types of muscles. Now cardiac muscles are only found in the heart. Only found in the heart. Now see, the walls of ventricles are much thicker than that of the atria. Have you seen the structure carefully? I have deliberately drawn this as a thicker wall and this as the thickest wall. Do you see? See here. This wall is the thickest. This wall is the thickest. So your left ventricle has the thickest wall. As compared to auricles, ventricles will have thicker wall, and out of the two ventricles, your left one will have the thickest of all the walls. Now, if I talk about the wall also, now see, this wall is made up of 3 layers. Yes, it is made up of 3 layers. See here, the outer one, this is known as epicardium. This is epicardium. Then we have a very thick layer called myocardium. This myocardium is actually made up of cardiac muscles. This is made up of cardiac muscles. Is that clear? And then the innermost layer, this is again made up of squamous epithelium. This is made up of squamous epithelium. So myocardium is going to vary in every part. So it is the myocardium or the layer of the cardiac muscles which is thickest in the left ventricle. Are these points clear? So there is this specialized cardiac musculature known as the nodal tissue. Now this is a very important question, guys. What is the SAN and AVN and all these Purkinje fibers made up of? They're not nerve fibers. They're modified muscle fibers. They are specialized cardiac musculature only. Be it SAN or AVN or bundle of hairs or whatever, they're all this. Now, where is nodal tissue present? Present in the right upper corner of right ventricle. And what do we call it? I've just told you, sino-auricular node, where the impulse generates, right? Another mass, see here, another mass of tissue is seen in the lower left corner of right, which is nothing but atrioventricular valve. I have already shown you the position of these two structures. Now, the bundle of nodal fibers and atrioventricular bundle continues from AVN. Now see, AV bundle is a third thing which passes through the atrioventricular septum to emerge. Let me show you with the help of the diagram. So, let's say this is your heart. So, I am going to just draw a quick structure here. See, upper right corner, this is your SAN. Lower left corner, this is your AVN. Yes, from AVN arises the AV bundle. So it enters into it is written, this enters into, where is it written? Continues the AV bundle which passes through the atrioventricular septum. So this is the atrioventricular septum. This is the AV bundle, right? And now once it enters here, it is going to split into 2 branches. It is going to split into 2 branches, right and left branch, and towards the apex it is going to break off into many fibers, and all these fibers are known as Purkinje fibers. So do you see how the impulse is going to flow? Purkinje fibers. So this is the right and left branch. So together, AV and the right and the left branch, these two things together are known as bundle of His. Are these points clear to all? Please tell me. And from here, see, right and left, and these branches give off rise to the Purkinje fibers. So they have the ability to generate action potential. The nodal musculature, we are talking about SAN. Without any external stimulus, it can generate the action potential. Hence, it is auto-excitable. And that is why our heart is also myogenic. So myo means muscle. This muscle of the heart will generate the impulse on its own. Is it clear? So the number of action potentials could be generated in a minute, vary at different parts. So SAN can generate around 70 to 75 potentials per minute. And this is exactly equal to your heart rate. As many times the SAN is going to produce impulse, your heart is going to beat. Is it clear? So important for initiating the heart. So brain can and hormones can increase the heart rate, but brain cannot stimulate your heart. So heart has to generate that beat on its own. Is it clear? So therefore it is called the pacemaker. Our heart normally beats around how many times? 70 to 75 times and average 72. So your heart rate is exactly equal to number of impulses from SAN is equal to your heart rate. Is that clear? Please tell me these points are clear to you all. And what is the sequence in which the impulses flow? So the SAN produces impulse and spreads it to both the atria, not one, both the atria. And from here it moves on to the AVN also. From AVN to AV bundle, then to the right and left branch, and then to the Purkinje fibers. Is that clear to all? Please tell me that these points are clear. Yes. Now we are going to talk about the very important aspect, which is cardiac cycle. Yes, guys, please tell me, are we clear until here? Let's have a quick break. Thumbs up break. Yes, please. Are we clear until here? Anyone with any doubt? Dinner break. You can eat your dinner if you want alongside, but I'll take dinner after this. Actually, what is meaning of that? I know it is. Oh, sorry. Okay. Bathroom break. Yes. Take a bathroom break, or a water break, a thumbs up break, or you can ask your doubts. All clear? Perfect. Preeti, pacemaker is another term given for SAN, because it is the SAN which decides how many times the heart is going to beat. So that is why we call it a pacemaker. Now all these structures, be it SAN or AVN or Purkinje fibers, all these structures are basically modified muscles, cardiac muscle fibers. They are modified cardiac muscle fibers, not nerve fibers. Please remember, it is already a PYQ. Is that clear? Now, so as many times, it is auto-excitable. So, there is an alarm set. So this is also a type of alarm that after every 0.8 seconds, SAN is going to produce an impulse automatically. That is why it is auto-excitable. Your brain and your hormones can influence your SAN, but they cannot initiate the impulse. Impulse has to be initiated by your SAN only. Is that clear? Shall we move on now? Yes, guys, shall we move on? So, Jyoti, I have been telling you side by side. See, I've told you already that your heart, the inner side of the heart, the inner lining, that is made up of squamous epithelium. All these muscle fibers, all these tissues, nodal tissues, they are your modified cardiac muscle fibers, right? So, these are the things. These are not even given in your structural organization. We are talking about it. Is that clear? SAN and AVN. Sankalp. Now listen, AVN is auriculoventricular node. What does SAN does? It produces an impulse and passes it on to both the atria, right? And also to the AVN. Now AVN is going to re-amplify that impulse and pass it on to towards the ventricles. Is that clear? So if SAN is a pacemaker, then AVN is also known as the pace setter. Is that clear? It is known as the pace setter. All right, all right. Is that clear? Okay. Let's have a quick water break. What if SAN was somewhere else other than the right atrium? Hex, what type of question is this? If it would have been somewhere else, then the body would have adapted to that. We would have never known to some other system. So this is just a fictitious question, a hypothetical world. Okay. Yes, guys. All right, all right. Moving on to the next now. Give me a quick break. Wait a second. Let me have a sip of water. Yes, guys. So are we back? Are we back here? How many chapters will you teach? We'll complete human physiology. Okay. All right, now moving on to the cardiac cycle. See here, talking about the cardiac cycle. Yes, please. So what is cardiac cycle? So cardiac cycle is how the heart beats, right? And that duration of all the events that are going to happen once the SAN produces the impulse. So all those set of events which will occur, which will follow after the SAN produces impulse, all those events are known as cardiac cycle. Right now, as a matter of fact, between two impulses produced by SAN, the time duration is 0.8 seconds. Okay, now let's see what happens during the 0.8 seconds here. All right, so I'm going to explain this with the help of my own flowchart. So let's focus here. So it is a cardiac cycle, right? Now we can divide this cardiac cycle or 0.8 seconds like this. Now see, so 0.8 seconds divided into 0.4 seconds and 0.1 second like this and 0.3 seconds like this. Now what happens during 0.4 seconds? The whole heart, both atria and ventricles, they are in the relaxed state. So we call the relaxed form of heart as diastole. So relaxation is known as diastole. Now please understand, when the whole heart is in the relaxed state, when the whole heart, both atria and ventricles are in the relaxed state, see, okay. Now what will happen? There is negative pressure in the heart. There is negative pressure inside the heart. Your TV and BV are open. TV, tricuspid and bicuspid valves are open. There is negative pressure inside the heart. So what will happen? So as compared to the heart, the superior vena cava or inferior vena cava, they will have positive pressure, they will have higher pressure. And the same is the case with pulmonary veins, they will have positive pressure. Is that clear? So blood from all these vessels are going to enter into the heart. So basically there is blood filling going to occur. Blood filling occurs, blood filling inside the heart during the joint diastole. Is that clear? So the blood which comes into the atria, now since TV and BV are open, it is also going to move into the ventricles. So basically 70% of the ventricular filling will take place during this phase. Now this ventricular filling is slow. This is slow. Is that clear? Please tell me. So this happens for 0.4 seconds and it allows the ventricles to fill up to 70%. That is 2/3 of the ventricles will be filled here. Now at the end of the 0.4 seconds, now see, SAN is going to produce impulse. SAN produces the impulse. Now as soon as SAN produces the impulse, both the atria will contract. Both the atria will contract. So atrial contraction occurs. That is why we call this phase as atrial systole. We call this as atrial systole. Yes or no? Please tell me. We call it atrial systole. Yes, atrial contraction occurs, right? Now, when the atria starts contracting, let's draw the heart again. See, when the atria starts contracting, so the blood which has been pouring into the atria, when the atria contracts, what will happen? The blood which is going into the ventricles, it will go faster. So, now rest of the 30% ventricular filling will occur here. Ventricular filling will occur here. But this ventricular filling is different from this. This was slow. Now this is going to be fast because your atria are squeezing. The atria are contracting from behind. Please tell me these points are clear. Please tell me these points are clear. Yes? Alright. Now this atrial contraction will only extend up to 0.1 seconds because while the atria are contracting and pushing the blood into the ventricles, SAN is also sending the impulse to AV node. Yes, and AV node will re-amplify and amplify the impulse and pass it on towards the ventricles. So when impulse is passed on to the ventricles, your ventricles will start contracting. Therefore, we call this phase as ventricular systole. Now please note, while your atria was contracting, your ventricles were relaxing. So during atrial systole, your ventricles were in the diastole condition. And now when the impulse has been passed on to the ventricles and ventricles contract, this also means now atria will go in a state of diastole. Is that clear? Okay, now atria has relaxed. Let's see what happens during the ventricular systole. Now as soon as, see here, so this is your heart, this is your heart. Now see carefully. Now as soon as the ventricles start contracting, the first thing which happens is your tricuspid and bicuspid valves close. Tricuspid and bicuspid valves close. Is it clear? TVBV close, right? Now when tricuspid and bicuspid valves will close here and they still start contracting, they still keep on contracting, what will happen? The blood which is all filled in the ventricles, this is going to put pressure. Now there we have opening of pulmonary artery and here we have opening of aorta, right? This is aorta and we have semilunar valves here. We have semilunar valves here, right? So once your tricuspid and bicuspid valves close and the ventricles keep on contracting, this is going to put pressure on semilunar valves. So your semilunar valves open. Is that clear? Semilunar valves will open, right? Now when the semilunar valves will open, the blood will be pushed out. Blood ejection takes place. Blood ejection means blood goes out. Where? From pulmonary artery it goes into the lungs and through aorta it goes into the body. Of course, this is the deoxygenated blood and this is the oxygenated blood. Please tell me these points are clear. Is that clear? Now, while the blood has been going out, the ventricular contraction is now also ending. Now towards the ventricular end of ventricular contraction, what will happen? As the pressure decreases now, see, when TVBV close, the pressure starts increasing. So there is positive pressure in the ventricles. And because of that, your SLV also open and blood goes out. Is that clear? Now, towards the end of the ventricular contraction, there is again negative pressure inside the heart. Negative pressure inside heart. Now, which will do what? Now, SLV will close, which had opened and blood ejection has taken place. Now, SLV are going to close, semilunar valves, both of the pulmonary artery and of the aorta. Is that clear? Now, when these close, what will happen? Now, TV and BV will open. Because of low pressure in the ventricles, TV and BV will open. And when these open, see, blood has been entering into the atria. During the ventricular contraction, when TV and BV were closed, blood was filling in the atria. So, as soon as the TV and BV open, now the blood from the atria, it rushes downwards. So, again, ventricular filling takes place. Is it clear? And because the blood rushes down, so this ventricular filling is also fast. This is also fast. Now, actually, this ventricular filling and this ventricular filling together makes 70%. But most of it occurs during the joint diastole because this occurs during the end of the ventricular systole. Please tell me these points are clear. Now, another point to be noticed here. Whenever a valve closes, it produces a sound. So, first sound is produced when TV and BV close, and this sound is known as, first heart sound is known as lub. This is known as lub. While when SLV close, this leads to the second sound, which is known as dub, So, do you see both the sounds are produced during the ventricular systole? First sound during the beginning and second sound during the end of the systole. Please tell me these points are clear. Please tell me these points are clear. Yes or no? Is that clear? Please tell me these points are clear. Right? So now do you see what all things and what all events are occurring during each phase? Yes or no? Do you see that? The events that are occurring during each phase. Can we move on now? So this is what has been given here and I have summarized it all. Now another point to be noticed. See, during the blood ejection, during ventricular systole, when I said blood ejects, blood ejection means blood goes out. During blood ejection, now see, the amount of blood that goes out of ventricles during one heartbeat, during one cardiac cycle or one heartbeat, this is known as stroke volume. This is known as stroke volume, and that is from during one cardiac cycle, and that too from each ventricle. Through each ventricle is known as stroke volume. So when the blood is ejecting out, 70 ml goes from the right ventricle, 70 ml from the left ventricle. This is called the stroke volume. Is it clear? Now, if I multiply stroke volume with the heart rate, that is number of beats per minute, this will give me cardiac output. Yes or no? So can you multiply 70 into 72? Can you get the right? It is around 5040 or 5000 ml or 5 liters of blood. And now different factors, they are going to affect the stroke volume as well as the heart rate. So as the heart rate changes, your cardiac output also changes. Please tell me these points are clear. Please tell me these points are clear. Yes? All right. Moving on. So if you increase the stroke volume or the heart rate, your cardiac output automatically increases. Now, point is, how do you measure your heart rate? You measure your heart rate through a special device known as stethoscope, and we can hear the two sounds. See, the first sound is lub due to the closure of TV and BV, and the second heart sound, which is dub, is due to the closure of semicircular valves, and these sounds are of clinical significance. So, is this point clear to everyone? Can we move on to now electrocardiograph? Now guys, those of you who are using the new NCERT know that NT has already rectified it to be electrocardiogram. It is not electrocardiograph, it is electrocardiogram. Now, what is the difference between gram and graph? Now, see, this electrocardiogram is basically that graphical representation is known as electrocardiogram. While electrocardiograph, so electrocardiograph is the machine. Electrocardiograph is a machine, and electrocardiogram, this is the graphical representation of electrical activity of heart during the cardiac cycle. Not the mechanical. It is not about when the heart contracts. No, it is about before that. It is actually why will heart contract. So it is about the electrical activity of the heart. Is that clear? Now, the standard ECG, to obtain the standard ECG, there has to be at least 3 electrical leads. There has to be at least 3 electrical leads. Where are they attached? One on each wrist and then to the left ankle. Is that clear? At least these. They might go up to 12 leads. Now listen. Now see. So for the detailed evaluation, we can use more leads also, one to the chest region and so on. Now let's see this typical or standard ECG. Now the standard ECG consists of certain waves. Do we see that? It is a P, Q, R, S and T wave, right? Now what does this P wave indicate, guys? The P wave indicates the depolarization of atria. This is not the same as atrial systole. It is the depolarization. So basically the impulse has reached the atria. Is that clear? It is the depolarization. It will be followed by atrial systole, but it is not the same as atrial systole. Systole will occur slightly after depolarization. Are you getting that? So this is the P wave. Now talking about QRS complex. So overall QRS complex is about depolarization of ventricles. Again, it is not the ventricular systole. It is the depolarization. It will be followed by ventricular systole slightly later. Is that clear? And then we have T wave. This is about repolarization. That means the ventricles are about to relax. Is that clear? It will be followed by ventricular diastole. Are you getting these points? Are you getting these points? Now see, there are some important lines written here. So P wave represent the excitation or depolarization leading to, it is not the same, it will lead to contraction of both the atria. Then QRS complex, we call it complex. It is about depolarization of ventricles. It will initiate the ventricular contraction later on. See, it is written very clearly, contraction starts shortly after Q wave and marks the beginning of the systole. Are you getting that? So it is electrical activity. We are not talking about the mechanical activity like how and when the heart contracts. That will occur followed. That will be followed by these waves. Now the T wave represents the return of ventricles from excited to normal state. That is repolarization. It marks the end of the systole. Now, the significance of Q wave, one more significance of QRS complex, the number of QRS is exactly equal to the number of heartbeats, or it is equal to the heart rate, number of heartbeats, or you can say it is equal to the heart rate. Please tell me. So you just count the QRS complexes and we get the number of heartbeats. See here. So counting the number of QRS complexes, one can determine the heartbeat rate. Is that clear? So any deviation from this shape indicates a possible disease or an abnormality. So it has a clinical significance. Please tell me all these points are clear. Is this clear? Anyone with any doubt? Anyone with any doubt? Is that clear? All right. Now moving on to double circulation. So we know about that blood enters the heart twice, first in the deoxygenated form and then in the oxygenated form. Now see, for these circulations, the blood flows through the blood vessels. So we have three main type of blood vessels. So what are the three basic terms? We have the types of blood vessels are, see, artery, vein and capillaries. So we have artery, veins and we have capillaries. Now let me tell you how these structures are present. See here. So in case of artery and vein, what is going to be the difference? So both artery and vein, let's see, both artery and vein, they have this outer known as tunica externa. Outer wall is known as tunica externa and it is made up of collagen. It is made up of collagen. Is that clear? Then inner to this we have this tunica media. Inner to this we have tunica media and it is quite thick in case of artery. In artery, this inner wall, that is tunica media, is quite thick. Is it clear? While in case of vein, this tunica media, that is the inner wall, middle wall, this is not very thick. This is not very thick. So this is tunica media. Is that clear? So do you see the difference? So externa are same in both arteries and veins, while media, it is thicker here and thinner here. Now this is going to bring the difference. So the inner wall, so the innermost wall, this is made up of squamous epithelium. See here, it is made up of squamous epithelium, like this. Very thin squamous epithelium found on the inner side, and this squamous epithelium layer is known as tunica interna. This thin is tunica interna. Now, as I said, tunica interna is made up of squamous epithelium. Is that clear? While the tunica media, guys, is made up of smooth muscles, involuntary muscles. These smooth muscles will allow the movement in blood vessels. They can contract, they can relax, right? Because of tunica media. Now, do you see the differences here? It is thick here and it is thinner here. Rest, both the membranes are same. Tunica interna and externa are same in artery and vein. Is that clear? Now, because of this, we see a narrow lumen here and we see a large lumen here, right? Because of the large lumen and because veins are very away from heart, there is low pressure also. There is low blood pressure here. So it can lead to backflow of the blood. That is why there are valves present all around in case of vein. While arteries are taking blood from heart away from the, carrying blood away from the heart, but they are nearer to the heart. So that is why there is high pressure, high blood pressure here. And we don't need any valves here. Valves absent. Please tell me these points are clear to all. Please tell me these points are clear to all. Is that clear? Yes, valves are absolutely present. Is this clear? All right. Now, if we talk about the third type of blood vessel, which is capillary. Now, this is the smallest one and has only one layer, and we've already discussed that capillaries are lined by squamous epithelium. And what do you call that squamous epithelium? We call it endothelium. The wall is so thin that it allows the exchange of substances through capillaries easily. Are these points clear to all? Yes or no? Then double circulation is also divided into, see, there are three, let me talk about the types of circulatory pathways. circulatory pathways. Now one, we have pulmonary circulation, right? Then we have systemic circulation. Also, we have portal circulation. And last not the least, we have cardiac or coronary circulation. Now, what is pulmonary circulation? So, pulmonary word is related to lungs. So, it is between heart and lungs, right? How? Heart and lungs. So, blood from the heart moves into the lungs. From left ventricle, it goes into the, from right ventricle, it goes into the lungs and it comes back into the heart in the left atria. So, this pathway is the pulmonary circulation. Systemic word is related to the body. So, between heart and the body. How? So, blood. So, this is the deoxygenated blood. This is the oxygenated blood. Now, see here. Between heart and the body. How? So, oxygenated blood from the left ventricle goes to the body. This is the oxygenated blood. And the body will return it in the form of deoxygenated blood in the right atrium. Please tell me these points are clear. Let me know in the chat, out of the pulmonary and the systemic circulation, which of these, guys, which of the two circulatory pathways are longer? Pulmonary or systemic? Let me know in the chat. Now, talking about portal circulation, it is between two organs, right? So, between two organs. For example, in humans, we have hepatic portal circulation, and hypophyseal portal circulation. While in frog, we have seen additional portal circulation, which is called renal portal circulation. Now see what happens here. Now in hepatic portal circulation, hepatic word is related to liver. So blood from intestine carrying lots of nutrients will not directly go to heart. It will first go into the liver and then move into the heart. So from one organ to another organ. Now the blood vessel which carries blood from one organ to another organ, this is called portal vein. And this vein is carrying blood towards the liver, so we call it hepatic portal vein. Is that clear? Hepatic portal vein. Then hypophyseal word is related to your pituitary. So blood from hypothalamus doesn't go back to the heart directly. It will first go into the anterior pituitary because it is bringing the releasing and the inhibitory hormones. And through anterior pituitary, it will go into the heart. Now this vein will be known as hypophyseal portal vein. Is it clear? Then renal portal, this is seen in frogs. We don't have renal portal circulation. So blood from the lower limbs goes into the kidney, and through kidney it will enter into the heart. So these are the different types of portal circulations. So between two organs. Is that clear? Now talking about the coronary circulation. Coronary word is related to heart. So when the blood comes out of heart, from heart to aorta, right, that is the left ventricle to aorta, then aorta will first supply some blood to the musculature of the heart. So musculature of the heart itself through arteries. So there are coronary arteries which will carry this blood to the coronary arteries, carrying blood to the musculature of heart, and it will return it back into the heart, but not the left but to the right atrium. So between heart, so here we have, see, heart, aorta, and then musculature, and going back through coronary veins. Is that clear? So this is between heart and around the musculature of heart. So these are the different types of circulatory pathways. Please tell me these points are clear to all. Anyone with any doubt? Yes, please. Anyone with any doubt until here? So this is the systematic plan of just pulmonary and systemic circulation. Are these points clear? Please let me know. And then finally, the regulation of cardiac activity. You see, the normal active heart is actually auto-regulated, है ना? Because it is intrinsically regulated by a specialized nodal tissue known as SAN. But the neural centers can also influence it. I've told that. Neural center in medulla oblongata, it can moderate the cardiac activity through autonomic nervous system. Is that clear? So the sympathetic nervous system is going to increase the heart rate and the strength of the ventricular contraction, and thereby the cardiac output will also increase, right? While the parasympathetic nervous system is going to decrease the heart rate and the speed of conduction, and thereby the cardiac output will also decrease. Is that clear? Other than neural control, there is hormonal regulation also. Now, how does hormonal regulation occur? Adrenal medullary hormones, that is adrenaline and non-adrenaline, both increase the cardiac output and the cardiac and the heart rate. Please tell me these points are clear to you all. Please tell me these points are clear to you all. Here we are talking about both adrenaline and noradrenaline. And they work along the sympathetic autonomic nervous system. They both increase the cardiac output. Please tell me these points are clear. And last not the least, we have disorders, guys. First of all, we have high blood pressure, that is hypertension. Now, hypertension is when your normal blood pressure is 120/80. This is a normal body blood pressure. Now, if the blood pressure, upper blood pressure, basically the systolic blood pressure is always 140 or above. So 140 or 90, this is called high blood pressure. Now, why is it such a problem? Because high blood pressure can damage your vital organs, right? Now, it shows hypertension. It is also known as hypertension lead to heart diseases and damage the organs like brain and kidney. That is why it has to be controlled. Then talking about coronary artery disease, this is also known as atherosclerosis. So basically what happens here, so this is your blood vessel, and because of excessive intake of cholesterol, so this is the cholesterol deposition. So cholesterol depositions are going to decrease the lumen, and eventually there will be blockage in the blood vessel. Is this point clear? Is that clear? So cholesterol deposition is going to cause blockage. Yes or no? Now this blockage can be fatal. This blockage can be fatal. So the vessels that supply the blood. Now in case this blockage occurs in coronary artery. See, this blockage will be harmful everywhere. But if blockage is in coronary artery, right? Coronary artery is the one which supplies blood to the heart musculature. I told you, heart muscle. So it is, if it is in the coronary artery, it is known as coronary artery disease. What will happen if it is in the coronary artery? So blood supply is blocked to the heart muscle. Blood supply blocked to heart muscle. Yes or no? When blood supply is blocked to the heart muscle, the heart muscle will not get oxygen, your heart muscle will not get blood, and it will die. So this will lead to another situation which is known as myocardial infarction. Myocardial infarction, that is death of the tissue because of no oxygen. Death of the tissue because of no oxygen is known as myocardial infarction. Please tell me that these points are clear to you all. So what does it say? So other than cholesterol, there will be deposits of calcium, fat, cholesterol and fibrous tissue. which makes the lumen of the arteries narrow. Which artery are we talking about, particularly if it is occurring in the coronary artery? Please tell me, are these points clear? Now, myocardial infarction means the heart muscle will die, right? What will happen? Your heart muscle is going to experience that pain, which is called angina, angina pectoris. So it is an acute chest pain when there is no enough oxygen reaching the heart muscle. And why does it occur? This occurs due to CAD, coronary artery disease, or myocardial infarction. Yes, it occurs in men and women of any age, but more common in the middle-aged people due to conditions that affect the blood flow. Are these points clear? Then talking about heart failure. See, now there are three different terms: heart failure, heart attack, and heart arrest, cardiac arrest. Understand these. See, what is heart failure? Heart failure means a state of heart when it is not pumping blood effectively enough to meet the needs. Is that clear? It is also known as congestive heart failure because it occurs due to congestion of the lungs. Is that clear? Now, talking about another, which is heart failure. Sorry, failure is not the same as cardiac arrest. Not going to talk about cardiac arrest. When you arrest someone, you stop that person from doing something. Yes. So, cardiac arrest is when heart stops beating suddenly. Your SAN fails to produce impulse. So, why does this happen? SAN fails to produce impulse. Now, if there is cardiac arrest, there is sudden collapse of the person, sudden collapse. Usually no comeback. Is that clear? Then there is heart attack. Heart attack is when the heart muscle suddenly gets damaged by inadequate blood supply. That is during the myocardial infarction. And this is when you feel angina, infarction. Please tell me all these points are clear to you. Please tell me all these points are clear to you. Is that clear? Is that clear to all? Please let me know. Thank you, Dhanush. So guys, by the way, I am going to take your leave now. We are going to complete human physiology tomorrow. My throat is really hurting. So before I get fever and we are not able to take the class tomorrow, I will be taking rest of the 4 chapters in tomorrow's session. Is it clear? There will be part 3 of zoology. We have exam on 21st, and side by side, guys, now we will start practicing these questions. There was this one student asking, where are these sessions taken? These question practice I am taking in special classes on Unacademy app, Learners app. These are absolutely free classes. You can follow me on Unacademy app and you'll get notified whenever I start the class. I'll inform you that on Sunday 9 o'clock, because tomorrow we'll have a marathon again. So Sunday we are not keeping a marathon. Sunday onwards we are going to practice the questions in our special classes. I'll keep 2 to 3 special classes on Sunday so that we can practice all the questions based on what we have studied till now. So please make sure that you watch and you attend those classes live. You solve those questions with me. This helps in making and having more focus and also understanding that what are the keywords that we have to focus on while solving the questions. Is that clear? And also guys, before I leave, I'll tell you once again, please subscribe to Prime membership, just ₹1, but you will be able to watch all the recorded lectures, take all the tests, and there will be so many questions that you need to practice before going for Re-NEET exam. So thank you so much. I'll see you tomorrow. Again, same time, 7 p.m. on Unacademy NEET English channel, and Sunday we'll be practicing the questions on Unacademy special classes on Unacademy app. Is that clear? Thank you so much, and I'm so sorry that I could not complete the human physiology, but I'm not really feeling well. So I'll be continuing with the next chapter of human physiology tomorrow, same time, 7 p.m. Thank you so much. Don't forget to join me tomorrow. Hit the bell icon. Don't forget to share this and like the session. So joining tomorrow at 7 p.m. with the rest of the human physiology. Bye-bye, guys.